This is a production of Cornell University.
Glad to have you all here at the 13th Annual Cornell Corteva Plant Breeding Symposium. I know this symposium has been the highlight for a lot of plant breeding graduate students here at Cornell over the years, and we're so glad that we get to be part of the larger Corteva Symposia series. Every year, our plant breeding graduate student organization, Synapsis, gets to vote on what our theme is. And this year, we chose something that was relevant to our students and that they thought was important. This year, our topic is plant breeding for novel systems and complex problems. We hope that this symposium will highlight some of the examples of how plant breeders are using out-of-the-box approaches or cutting-edge methods to adapt crops to unique systems. This symposium would not be possible without this Synapsis Symposium Planning Committee. I'd like to take a moment to thank my co-chair, Sam, and also our other committee members, Tim, Vaswati, and Shrikant, who unfortunately couldn't be here today. If throughout the symposium you have any questions, you can try to find one of us and we'll try to help you. I'd also like to thank Cornell for their continued support of this symposium, and also BTI for hosting us again. And then, of course, Corteva for founding the series and continuing to fund us and support us.
I wanted to thank the organizing committee for inviting me to come speak. I've been involved with the University of Minnesota's symposium series for 3 years, and so it's really come full circle to be an invited speaker here. My talk today will be a little bit different than some of the others in the fact that this is volunteer work that myself and a large team of graduate students have done. And so this is just some of the names of the second and third cohort of our group. Really want to emphasize that this is a team-based activity. A little bit of background about where is the need, where's the objective of our group. And so in 2016, this article was published about Minneapolis Farmers Market. I might let you guys read the first quote there. And I would summarize it in saying that there is a large need for these traditional African vegetables, that there are community members driving up to 8 hours away for these specific vegetables. And even then, they're not getting everything that they have. They're running out on time. And so I might pose a question to you. If you move to a new country, what will you do if you cannot find broccoli or carrots? You could adapt to the local cultural vegetables, or maybe you could bring your own vegetables here. And so I might pose a greater question to the group here. Are we as plant breeders, are you all as plant breeders, meeting the needs and demands in a more socially equitable way? And so to give a little background about where we are in Minnesota as a whole, within Minnesota, around 9% of the residents were not born in the United States. Most of those live within the Twin Cities, and of those, around 150,000 list Africa as a region of origin. And so our group is really focusing on West and East Africa, communities from these regions. And within the pie chart up here, I might highlight, we have large populations within Twin Cities from Nigeria, Liberia, more in the West Africa, more in East Africa, Kenya, and Somalia. And this figure on the bottom just shows that over time, within the last recent 5-10 years, the proportion of foreign-born people within Minnesota from Africa is really increasing the most. And so to introduce our team, our main mission is to bring together researchers, growers, and students to develop culturally relevant food plants towards Minnesota. This is a picture of our group, of the current iteration, the current cohort. A lot of our group has really been modeled after Scope out of UC Davis. This more participatory volunteer work of graduate students. One of our main goals is to eventually release culturally relevant vegetables that are specifically adapted to a Minnesota climate. So a shorter growing season, cooler evenings, cooler springs. And our group was started in 2021. And this is a picture of it. And just to highlight, this is all volunteer work. So some of our main collaborators that we work with, that we have learned in a mutual learning environment. So Chidi Chibozi is our main one. He is with the Hennepin County Master Gardeners. That's one of the main counties within the Twin Cities. And he is in charge of the DEI within the Master Gardeners group there. Other collaborators highlight would be Ngozi, Natalie Hoydal is with UMN Extension, and Zachary Page, who runs a nonprofit seed production business. So we are partnering with them to actually produce seeds to get to the community. To give a little broad highlight about what participatory plant breeding is, or even in our context, what it is. So some of the main questions would be, who defines the breeding goals? Sometimes the scientists would, sometimes the community would. And so in our vision of participatory plant breeding, we're not the ones designing the goals. We do not know these vegetables intrinsically, and so we're working with, not for, the community to develop that. And so Frank talked about G by E by M. We really think about this as G by E by C, so the context, the role that people play within developing varieties. And so our target environment would be home gardeners or small-scale producers within the greater Twin Cities area. And just to highlight more about that context can even be subdivided into regional specificity. So we have certain vegetables that, depending if you're in East and West Africa, the eiotypes are quite drastic, quite opposing. And then even when you dive down, what we are really doing is more participatory variety selection. And so participatory plant breeding, where the community is involved in every step of the breeding program, we're not there yet. We're really walking before we are running. And so we are screening diverse germplasm. We are selecting varieties with the eventual goal to have that variety have these idiotypes of form a more traditional breeding pipeline. And so some of the ways that participatory plant breeding does really well, is really ideal for, is in these highly heterogeneous environments. Okay, home gardeners within the Twin Cities, every garden will have a different set of conditions. When advanced technologies are limited, or I might even change that, not limited, but not wanted. So a lot of our communities really do not want hybrid vegetables, do not want genetically modified in any capacity. And lastly, to a broad range of stakeholders. So when our group first started, we did a situational analysis, okay? Where we are, what are the priorities? And so in our priority setting, we said we really want to focus on leafy greens. There are a large number of African vegetables out there. There are fruit that have an okay ability to ship. There are small grains that should have a very good ability to ship, but we really focus on these leafy greens, these vegetables that really need to be produced locally, that we have seen that need for within the farmers markets. And one of the ways we did that is look, okay, are there existing varieties? For some of our species, the answer is no. You cannot get these seeds anywhere online in the US. And so in sort of the context of how they're used, they're always cooked. And that's one of the weaknesses that we're trying to develop, is building our relationships with home cooks. And so looking at the stakeholder preference, we have hosted several open houses and farmer days where we ask the people, these communities, what do you want? Most of this learning is done through verbal communication. And so these are some of the highlights of what they are interested in. They're not interested in hybrid vegetables. They want to be able to save their own seed. They are not interested in genetically engineered, which most vegetables are not. And this third one here is currently a large challenge for us to try and overcome. There's no IP rights, that you really don't own a cultural staple. And so a lot of our work really aligns close with the open source seed initiative. And lastly, quality is much more preferred over yield, that these food aspects, the taste, the flavor, the texture of these vegetables is so intrinsically important. And lastly, there are greater cultural functions in some of these vegetables. On the right up here is bitter leaf. So bitter leaf is a tree-forming Asteraceae, one of the most bitter leaves you can ever taste, really used in a medicinal way. And so what we're trying to bring to the table as volunteers in this mutual learning aspect is that we have access to resources at the University of Minnesota. We have greenhouses, we have field equipment, we have the space, we have the volunteer capacity to really form this as a center. And within the last couple of years, we had a community garden and a demo garden on campus. These are in organically managed fields on campus. And lastly, we have access to genetic material. So we talked about before, there's really no, in some instances, ability to order these seeds online. As researchers, we have access to GRIN, but GRIN is not available to the general public. And so in some ways, we're just serving as a repository to that seed, as well as other sources of germplasm, such as the World Vegetable Center in Taiwan. And some cases, we act as seed multiplication. We had a farmer give to us some African eggplant seed. He actually lost his entire collection, and we still had a backup of that, and we're able to reprovide him some of that seed. And we talked about mutual learning a little bit, and our eventual goal is to release a variety. We have come really close with one of our crops called jute mallow, but I'll highlight that in a little bit. So going a little bit about how do we get there? How do we as volunteers actually accomplish this? Most of this is relationship building. Relationship building is integral. It's slow, but it's effective. So in the last 4 years, we have devoted 90% of our time on this relationship building, less on the actual breeding of these vegetables. Most of this is done by word of mouth. It is not our place to spam out emails to a long listserv. We are interested in the public that actually, the percentage of the communities that actually want to work with us. So within that, there's a strong emphasis on participation of knowledge-rich stakeholders, and GOZI is one of them, as well as Chidi. And recognizing that a small subset do not want our involvement, that in the past, University of Minnesota has misappropriated some species, and that resentment is very real. And so we are working with the communities that are interested in working with us. And so some of the ways that we do that, we provide seeds to community gardens throughout the Twin Cities. We are really narrowing our focus to a smaller subset within the greater African leafy vegetables. And in some cases, there is just a real lack of available accessions. And so we have a small project work on creating genetic diversity through mutagenesis. We send a small subset of seed to the nuclear reactor at UC Davis for mutagenesis diversity generation. And so how does our group actually accomplish these building relationships within the community? We host a very large open house every year. We have a booth at the Organic Specialty Field Day. And just a lot of reflective looking back, is our language, is our goals aligned properly? We get feedback from DEI, we participate in conferences. 2 years ago, we wrote a guide to actually growing these emerging vegetables, and it won an award at the American Society of Horticulture meetings. And we also are out with the community in these farmers market. I believe we've attended 6 farmers markets last year, just getting our name out, spreading that word of mouth. And so I want to highlight one in particular, our field day. So our field day was a great success. We had around 125 participants come to our plot on campus. And so one of the ways that we connect with people is over food. Food is so essential, so part of the cultural identity. So at the top left, we had a potluck style dinner before we actually went to the field. People, the community were encouraged to bring food. We had taste testing on the left here of all of our varieties, or this variety selection. At the end of the day, we had a giving garden. People were able to take home large quantities of these vegetables to try and cook for themselves. And here's just some pictures of the actual field day. And so very briefly talking about some of our focus species, we were working on jute mallow. So jute mallow is the same plant made for jute fiber, but in a vegetable crop. So these are different germplasm pool as a leafy green. Ethiopian mustard, so Brassica carinata, also an oilseed, but we are really focusing on the vegetable aspect of that. Cleome, also known as spider wisp, and amaranth. And just to highlight a little bit about some of the idiotypes that we're working with, some of these, we want them to flower earlier. So for jute mallow, really will not flower in time for Minnesota. They'll produce seed pods, but they won't mature. And so these are real tangible goals that we can increase the flowering time. Or conversely, with Ethiopian mustard, we want to delay that flowering time. It bolts too soon. It's a leafy vegetable. It makes great seed production, but as soon as it bolts, as soon as it flowers, the leaves turn more bitter. So these are aspects that are tangible, we feel, within our focus species. So to highlight some of the current challenges that we have, one of the main challenges we ran into is just the common names. So common names are useful, but they're clunky. And recognizing that some common names come from a very challenging history. So for example, cowpea is the name given by slave owners, by colonizers, that demotes a crop for humans to nothing more than fodder for livestock. And within the greater scientific community, cowpea is still the main word used. And so we use that while recognizing that different language is really needed. Moving on to some of the other challenges, some of our focus species have 20+ common names. When we are writing our grower guide, how do we choose what name goes on this guide without leaving out certain communities? And recognizing that a real subset of our audience does not trust university researchers, and being aware of that and respecting that, respecting boundaries. One of our challenges really is the lack of feedback from home cooks. So we are able to get this vegetable out to the community, but we have really had a challenge actually talking with the people cooking these, the actual final end user. And so that's one of the things we hope to improve in the future. And lastly, making selections on flavor. This is a picture of me trying bitterball. So bitterball is this very bitter eggplant. I really do not like bitter flavors, and that's authentic. It's important to be realistic, to embrace these. But within the greater goal of participatory plant breeding, we are not the ones making selections. We are not applying a Western sense of flavor. So a lot of some general themes to these African vegetables is bitterness. They like it to be bitter. They want it to be more bitter, as well as texture. The slimy, mucilaginous aspect, wherein American would cook okra, we would hide that. We would bread that. A lot of our communities will make a soup out of okra to enhance that slimy texture. Some of the things that we've learned, so language matters, even just within what we call our group. So our working name for a while was vegetable breeding for immigrant farmers. And we really changed that. We recognize there is a power dynamic when you say we are working for. So we really strongly changed that. We're working with communities. And now our new name is the Community Plant Breeding Team. And I should say this is an outreach arm of the Plant Breeding Center under the guidance of Rex Bernardo. Even just some of our language around the word immigrants. So we are working with people that are second generation, third generation from immigrants. We are working with people that might be on refugee status. And so some of our language would be moving more towards the African diaspora instead of using the word immigrants. Respect boundaries, very important and challenging at the same time to learn about that and to maybe try and heal some of these perceptions about the University of Minnesota. Another thing that we learned, don't take things at face value. So some of the scientific publications out there that you would read would say this species is for, or this eutyotype, this germplasm pool is for vegetable consumption. This is for biofuel, for fiber. Well, when actually talking with the community, it turns out some of the published articles are wrong or flip-flopped. So really highlighting the importance of that oral knowledge, listening to the community, learning from, and learning with. So with that mutual learning, we are learning so much more, so much about these vegetables. We are not pretending to be experts. We're able to share some of our knowledge in plant science while still learning at the same time. And I really want to highlight how rewarding that has been. So I work in the soybean breeding lab. This research, this volunteer work has really changed my perspective and just how we interact with stakeholders and the diverse stakeholders and meeting different needs. And sometimes stakeholders have contrasting endotypes and how we can bring this knowledge to our research, to my research. And then so what's next? So we are really highlighting this year, reaching out to home cooks, sort of quantifying that flavor and texture of these vegetables within our available diverse accessions. We hope to release a variety in the near future. So we have one for jute mallow. So over 4 years, we have been selecting for early and earlier flowering, and we have seen a gradual increase in flowering time. And so we believe that is ready to release the seeds. But one of the main aspects here is, okay, we are not going to monetize this. We're going to freely release this. There might be a small cost for the production of the seed, and that's where we're partnering with North Circle Seed for that. But we're really viewing ourselves not to monetize off of this work. Our outreach, so we are developing more ways to quantify this information. Some of these communities have expressed to us a real challenge in conveying their knowledge to the next generation, to their children. And so we're helping write down this knowledge, make handouts that are able to be given to the interested people, as well as education. So we work really closely with the Master Gardeners community. They are required to have continuing education credits every year. And so we act as a qualified provider of those credits, as well as increasing our community engagement. Just 2 weeks ago, I was at our local Natural History Museum talking about our work, talking about the community, and talking with just other non-target community members that are really interested in the work we're doing. And so we invited them to our field day to come and taste these vegetables, to see them with your own eyes, to feel them with your own hands. And lastly, we're really sort of outsourcing, working with a third party of North Circle Seeds to actually be the seed supplier. And so within this, I want to acknowledge our group. All this work is done at the St. Paul Research Farm with a lot of helps thanks to Andy and Jason. This is on Student Organic Farm. A lot of our students here have no experience working on organic farm, and so we really relied upon their knowledge of organic practices. So big thanks to Tori Dahl, as well as within the Hennepin County Master Gardeners. They provide probably half the labor force required for this field. It is a large amount of work. On the right here, you can see we lay drip tape, landscape fabric. Most of the things that we do are transplanted. And it is important to acknowledge that this took place on the University of Minnesota land, which is in the traditional homelands of the Dakota people. And lastly, for funding, we received some of our funding from our College of Ag, which is CFANS, and the Plant Breeding Center. And so with that, I thank you and open time for questions. Yeah, that's a great question. So most of this has been active learning. We sit down as a group, we learn together. And in some ways we make mistakes. So a lot of our early work, particularly related to language, we have learned, okay, this is not an ideal way. No, I wish we actually had better teaching classes for participatory plant breeding. I do see that as maybe a weakness within the University of Minnesota. Yeah, that is an excellent concept and really a major challenge of us. So we are student volunteers. Our average volunteer length would be 2.5 years. I'm part of the second cohort. I am transitioning off at the end of this month, actually. And so one of the ways we do that is we have an onboarding and offboarding documentation. But even then, there are some limitations to that. So within our group, we have assigned positions. And before one person offboards, you need to find a replacement. And so we're working with that, but we see that maybe a long-term technician would be incredibly beneficial to provide some continuity across time. Yeah. And so Rex is there, but Rex is a very hands-off advisor. And so if anyone is interested in collaborating, writing grants to fund technicians, we'd be really interested in that.
How does it get started?
Yeah, that's a great question. I was not there during the start of this, so I'm not sure how much I can provide. I know that Rex was really the driving force behind this. He saw the need through the article, and through that really devoted a large amount of funding from the Plant Breeding Center to work with this. To more ask your question about how do we connect with the community in a respectful way, it's just really slow. Word of mouth, being authentic to yourself, going out there, and really teaching people, okay, it's really important to respect boundaries. It is challenging sometimes to hear that a subset of people do not value work, protest your work, and being respectful is part of that learning process. It's really provided a great amount of experiences to us at the University of Minnesota.
Thanks. I'm in my notes here. I have a couple times over the cool. So just to follow up on Solveig's question, I think it'd be really neat if there was an oral history of your project. I don't know if that's could be worse. And then the other thing I just wanted to say was I love the fact that you missed the actual that were taken from this role. I think we should. I mean, we have lovely land acknowledgement also. Ties it back to the value that was taken. Thank you.
Yeah, and it's important to recognize that because Cornell took the most land. Yeah. Yeah, that's a great question. It is a big challenge. Within that Open Sea Source Initiative, there is a very weak verbal agreement, just that, okay, if you receive seed, you will not develop a product. You will not commercialize this germplasm. But it has been a challenge working with some of these, even just like the World Vegetable Center. There is an MTA associated with that. There is an exception for community members, but if you're not a community member, it is our responsibility if we give you seed to pass on that MTA. And so just that fact of passing on MTA can sometimes be very prohibited. It's a challenge that we're working with. Just yesterday, I was talking with maybe acquiring some cassava cuttings, and well, there's MTA process there. It requires going through our lawyers. That's a 6-month process. and being aware of that.
How did it to your
Mm.
research and also hobbies like that? Like what strategies are you using? It's very important work, and I'm just curious.
That's a great question. And if you have any advice, I'm definitely open because it is a challenge. And so Rex really advises our students, okay, make sure your research comes first. But with this work that we're doing, it's so fun. It's so easy to just, oh, I worked 5 hours on this. It's a work in progress. It's important to juggle that, to respect that. And I guess maybe just ultimately relying upon our team. We have a team of 12 to 15 people, and it's important to delegate those activities, to trust them in doing the work, and just work collaboratively. Yeah.
For your participatory variety selection and engagement community,
Mhm.
how do you guys organize documentation of your preferences or whatnot, and so it's survey form, or you just do this conversationally and
Yeah, that's a great question. So within our field day 2 years ago, we had the idea of doing surveys. We printed out surveys, we handed them out. 1 of 80 people filled out a survey. And so one of the best ways to connect with our community really is verbally. And so within our large team, we delegate people, okay, go out and talk to attendees and remember what they said and write down afterwards. There might be more efficient ways to do that, but that is where we're at now. That's a great question. It is a big challenge of us. We recognize that. We try and say, okay, hybrids are more productive. There is a benefit of that, but also maybe change our viewpoint to see where they come from. They really want to save these seeds year after year. In a hybrid setting, that would not be possible. Within hybrid seed, there's a greater upfront cost to some of these, which might be prohibited to certain community members. And so we, in this mutual learning, we're conveying that. While also learning from them their preferences and respecting them.
And we're going to be taking a 15-minute coffee break. Please be back here at 10:50 for our next speaker. Thank you all so much.
Welcome back, everyone. Hope you enjoyed some refreshments and some coffee. I'm really honored to introduce one of our own, Solveig Hanson here. Solveig is part of Dr. Ginny Moore's lab, where she leads the National Cover Crop Breeding Network. Solveig got her PhD from the University of Wisconsin and completed a postdoc at the University of British Columbia. She also, in her past, co-owned and operated a vegetable farm up in Iowa. And so Solveig is well-versed in genetics, genomics, production agriculture, and participatory research. And she is here to present us her talk toward 40 million cover cropped acres, breeding fall-sown cover crops for ecosystem services and farm system compatibility. Let's welcome Solveig.
Thank you for having me here today. I'm excited to represent the Cover Crop Breeding Network and to add our stories to this conversation about breeding for novel and complex systems. I've titled this talk to evoke a big vision, which is the same way that the Cover Crop Breeding Network began its work back in 2015. So I'm going to start by unpacking that vision. Why cover crops? Why 40 million acres? And why toward, as in why aren't we there yet? Then I'll talk about the role of breeding in cover crop adoption, what makes cover crop systems novel and unique and therefore apt to talk about today. I'll discuss our breeding pipeline and then some crop-specific breeding goals. So soft seed in vetch, cold tolerance in winter pea, and allelopathy in rye. And then I'll talk about some frontiers that we hope will include many million more cover cropped acres. So first, why cover crops? Cover crops offer many benefits that become obvious when you look at pictures like this. So on the left, there's a soybean field that has had some early season erosion, and then a cover cropped soybean field that has held its soil. On the right, you see some soil from a Concord grape orchard in Western New York, and you can see the difference in soil structure after 10 years of cover cropping. Among about 550 farmers who responded to the latest 2022-23 National Cover Crop Survey, people cited many reasons for using cover crops, but most of them revolved around soil health. So building soil structure, increasing water infiltration, reducing nitrogen leaching. Other benefits include creating pollinator habitat, and weed suppression, things like that. Cover crops can be sown in the spring or summer to cover soil until fall, or sown in the fall to cover soil over winter, or to winter kill. Fall sowing takes place after cash crop harvest. So between mid-September and late October in this area, that planting window is often quite narrow and in competition with a lot of important harvest tasks. Cover crop establishment is generally best when it's drilled or planted directly into the soil, but sometimes it's also flown on or interseeded into standing crops to mitigate that late season time crunch. Fall sown cover crops are terminated in several ways. They can be tilled, they can be rolled with that a roller like this big burly one, or they can be sprayed down. So cover crops provide important ecosystem services. So why do I specify 40 million acres? The 2017 USDA Census of Agriculture cites a little over 15 million cover crop acres in production. We are still awaiting the comparable figure from 2022, but we see from these annual national cover crop surveys a steady increase in the number of cover crop acres per respondent. Cover crop incentive programs are active and growing at federal, state, and private or corporate levels. And recent work from Matt Ryan's group here at Cornell showed that even after ceasing participation in these incentive programs, cover crop acres remained about a third higher on farms that had participated. So once people see what cover cropping can do to their soil, they want to, at least some of them want to keep doing it. So why 40 million? 40 million has been designated as an ambitious but reachable goal by the National Cover Crop Variety Development Project, a CAP grant that we are a part of. And finally, why toward? Why aren't we there yet? So cover cropping takes time to learn, and it may not be right for every situation. The issues here were top of mind for about 150 growers who responded to the National Cover Cropping Survey, saying they weren't using cover crops at this time. And these can be legitimate concerns, especially for growers who are earlier in their cover cropping experience. So some farms do experience economic benefits, say, from lower fertilizer or herbicide costs, but not every farm and not every year. But the items that I've starred here, achieving a good stand, mitigating cover crop weediness, and helping with the financial cost benefit analysis of using cover crops are things we can work on through breeding. To identify breeding goals, the Cover Crop Breeding Network back in 2016-17 carried out a survey of over 400 farmers and asked them what traits they thought were most important for the crops we plan to work on. The most frequently cited was nitrogen fixation, which we have investigated and found correlates very highly with biomass. Biomass is an important goal anyway, because in combination with early vigor, it helps with weed competition and all of those soil health benefits that come with decomposition from that cover crop residue into the soil. Winter hardiness is a big goal because perhaps obviously it is necessary for spring stands in places with cold winters. And finally, we didn't ask on this survey about seed yield because it's not a farmer-facing trait necessarily, but we know that that's important to make cover crop seed affordable for farmers. So what makes breeding cover crops novel and complex? First, crop domestication usually involves elimination of seed dormancy or hard seed that fails to imbibe water right after planting to emerge in the first season. Crimson clover and hairy vetch, two of our crops, still have substantial proportions of hard seed. Hairy vetch is also prone to pod dehiscence or pod shatter, which makes seed production less profitable and exacerbates that weediness issue that's caused by the hard seed. Even for crops like winter pea and cereal rye that have undergone domestication, most breeding thus far has been done for either food quality or for forage quality, not for cover crop use per se. In addition, the cover crop seed system is relatively less developed and less consolidated than major cash crop seed systems. So this is a figure from a CCB network paper that talks about 3 tiers of cover crop seed companies. So tier 1 are companies that sell only to other businesses, symbolized by a semi-truck. Tier 3 sell directly to farmers, symbolized by a pickup truck. And the tier 2 companies do some of each. As of 2021, over 40 companies were in tier 1 or 2, so doing some or all wholesale sales of cover crop seed to a variety of different regions and farm markets. So there are a lot of possible points of entry for cover crop breeders to get varieties into the marketplace, but it's really important to know what region different seed companies target and what cropping systems they target. And last, I'll mention that consumer farmer bubble down there is a really vibrant community of practitioners and researchers. I really enjoy coming from the organic farming side, that it's a combination of organic and conventional farmers and a really productive and creative exchange of ideas. Finally, most cover crop seed that is used right now is variety not stated, or even farmer grown and unlabeled seed. The advantage to VNS seed is that it's widely available and inexpensive. The disadvantage is that you don't know exactly what you're getting. And if you really like how it works, you can't necessarily go back and get more of the same for a following year. So VNS seed is better than nothing. It's important to get soil covered. That's the first priority. But as breeders, our challenge is, can we improve cover crops enough that it's worth growers money to buy a name variety to increase their chances of success with cover cropping? We're also interested in supporting a skilled informal seed system, or seed growing, selection, and trade among farmers. In addition to a formal seed system that's directed through seed companies, we feel we need to realize that because cover crops offer a long-term ecological benefit, but not necessarily a short-term economic benefit, farmers will have different budgetary constraints depending on their cropping system and overall situation. So we need to provide multiple avenues for getting high-quality cover crop seed into the marketplace and onto fields. So the novelty, the opportunity, and the challenge of breeding for cover crop systems are that we're working with species that have undergone less breeding work. We're working with a younger seed industry, and we want to support both formal and informal seed systems. And that fact that cover crops offer lower short-term economic value, leading to competition or coexistence with VNS and farmer-grown seed. So in response to this challenge, the Cover Crop Breeding Network was founded back in 2015 as a collaboration among universities, ARS research programs, and NRCS plant material centers. It has grown to include many sites. This year we have 9 breeding sites, 4 farmer breeders, and 15 trial sites across the country. In this first nationwide public cover crop breeding effort, the group started by working on 3 of the most commonly sown winter annual legumes, winter pea, hairy vetch, and crimson clover. About 2 years, we started working in earnest on rye, and then we added brassicas this year, focusing on winter canola. So each of these crops has a leader who is responsible for breeding pipeline design and centralized analysis of data from these various trials and nurseries. And notably, Cornell serves as the hub for the Cover Crop Breeding Network. My boss, Jenny Moore, is the National Program Director for the Cover Crop Breeding Network. And as coordinator, I get to work with over 50 technicians, researchers, students, farmer collaborators across the country to make this thing run. And to note, I get to be the one talking up here today, but the stories I'm telling wouldn't be the same stories without contributions from all these people. So as I've mentioned, in all our crops, we're breeding for biomass, vigor, and winter survival. We also have some crop-specific goals. I'll talk specifically about soft seed and vetch, cold tolerance and freeze-thaw tolerance in winter pea, and both allelopathy and cold tolerance in rye. This is our, you'll notice the question mark under the brassicas. This is our very first year looking at brassicas. So in collaboration with Mike Stam at Kansas State University, who's just supplied some germplasm from his program that he feels could have promise for cover crop applications. So we're looking at it, and once we see what's there, we'll figure out where we need to go with the breeding program. For the legumes and rye, our pipeline starts with breeding nurseries like these, where we note and select single plants. For the legumes, which we've been working on for the longest time, the next step is advanced line trials. So these are 15-foot plots flanked by triticale to simulate that competition with a small grain in a mix, which is how they're commonly sown. We take vigor ratings in fall and spring, percent stand in fall and spring to gauge winter survival, and then biomass harvest in a reasonably appropriate time for each site. The best advanced line trials go into mixed legume triticale plots to more accurately reflect the way they would be used in the field. And then concurrently with both levels of advanced line trials, we run seed yield trials so that we can either cull lines with low seed yield despite good field performance, or identify lines with exceptional seed yield to advance them in the process or use them as parents. And then putting it all together, to breed for cover crop performance, we start with germplasm sourcing and or development, which I'll describe in some of these crop specific stories. We selected in nurseries and then choose the best lines for advanced line trialing. The trial plots scale up as the lines do increasingly well over years of testing. Concurrently, we run these seed yield trials, first in one location. Corvallis, Oregon is in a major seed producing region, so it's kind of an ideal seed production environment for many crops. And then we expand to more environments to try to identify additional seed production regions as lines advance. And then finally, as we get to commercialization, some stories, as in the vetch story I'm about to tell, we're satisfied with data from our multi-site trials and wanted to move directly to release. Some want to test material in their own trials. So those are the general routes that get to commercial availability. So we will start with hairy vetch. Vetch is the best overwinter annual legume for both winter survival and nitrogen fixation, but it has a reputation, as some of you might know. It can get massive and viney, which is great if you have the equipment and the timing to kill it when it needs to be killed. It is less great if you don't have either of those things. And it has that fraction of hard seed and is prone to pod shatter, which allows vetch to become a weed in certain cropping systems. Vetch is an insect pollinated, outcrossing, highly heterozygous species, such that the varieties I talk about, or even the lines, are open pollinated. The CCB vetch breeding program has been led by Lisa Kissen-Kusick and Heathcliff Ryday at the ARS Dairy Forage Research Unit in Wisconsin. They started with vetch at 6 nurseries in 2016. They had a wide germplasm search, pulling in germplasm from Russia, Ukraine, Serbia, China, Iran, France, breeding populations from the upper Midwest, a couple of VNS varieties from seed companies, even a farmer-maintained landrace from North Dakota. Their goal was to find the most genetic diversity possible in the hope that these variation for these traits they cared about, winter hardiness, biomass, vigor, early flowering, would be in there. Nurseries are planted, replicated and space planted in multiple locations using the vigor and winter survival data provided by each location. Lisa selects among families, then within families for the best plants. Seed is harvested from every plant, and then a balanced bulk of seed is made from all of the harvested plants to comprise the line that is then entered into advanced line trials. So this is a fairly traditional forage breeding process, but it has been successful in achieving a couple of upcoming releases. This is the first one. This will be available through Mountain View Seeds, which is a wholesale or tier 1 distributor starting in 2025, if all things go well. This population was selected in our 2017 early maturing vetch nursery in North Carolina. The table here is composed of data from about 50 years or 50 site years of advanced line trials, and all of the numbers are represented as a percentage of the best check, which is AU merit. So you'll see that biomass, winter survival, and early flowering are comparable to the best check, but what really stands out is higher seed yield and much higher adaptation. So CCB nitrous is predicted to be in the top 3 in biomass for 3 times more environments than AU merit. So this is the power of this broad testing network that hasn't been available before for cover crops to detect varieties that are broadly adapted, at least for biomass, which is the trait we're talking about, and then get them out there to seed companies. Our next release that's upcoming is with Albert Lee Seeds, which is a tier 2 seed company, so selling wholesale and also directly to farmers. It focuses on the upper Midwest and Northeast. So we, at seed companies' request, we do separate analyses focusing on various regions so that we can give them results most relevant to their consumers. This line was actually selected here at Cornell in 2018. And it's very well adapted for winter survival and biomass in this sales area. In addition, seed yield is about 20% better than AU Merit. So you can see that seed yield is something that seed companies are gravitating toward, not mysteriously. Our multi-environment trials have also lent some insights regarding trait relationships in hairy vetch. So after analyzing data from about 50 site year environments, we found high and very significant correlations between several of our most important traits. So fall vigor, spring vigor, biomass. We also found that biomass correlates moderately with winter survival. And all of this is great because we don't have to make big trade-offs between these important traits. The only trade-off we found is in yellow there. So that's winter survival and early vigor. So we could face a challenge selecting for both early flowering and winter survival. Lisa Kisin-Kusik, who led this research, also analyzed environmental, genetic, and G by E components of each trait. And you'll notice that the blue bar, which is the environmental portion, is vastly larger than the pink and orange portions, which are the genetic and G by E effects. But she then overlaid or associated weather and soil factors and found salient drivers of various components for various traits. So for example, the environmental component of freezing degree of winter survival is driven by freezing degree days without snow cover, which isn't really a surprise to any of us who have worked with overwintering crops. Snow protects crops, so if you have snow cover, it can get really cold and more crops will emerge undamaged. But when you have those really cold temperatures without snow protection, that's when a lot of crops struggle. We also saw that the G by E component of biomass was related to sand content. So lines having variable performance, some doing very well in sandy soils, some doing very poorly and vice versa. So these insights, this analysis is done. We're doing similar ones for winter pea and crimson clover. These help us better define our trialing environments and our breeding environments so that we can identify whether we call them mega environments or environmental groups that are relevant to breed together to constrain genetic variation in strategic ways instead of trying to breed for the whole environment for traits where G by E is highly influential. In addition, thanks to more work from the USDA Dairy Forage Research Center in Wisconsin, we can leverage a new reference genome for hairy vetch. Hairy vetch is in the Vicia genus of the Fabaceae family. Fabaceae also includes peas and beans and many more species, genuses. The first chromosome level Vicia genus was for common vetch, or Vicia sativa. That was published just in 2022. But that species is self-pollinating and highly homozygous. The vetch genome was assembled just this past year in 2023 from long-read sequences and is just published. It used a clonally propagated individual of AU Merit, which you remember was the best check from our field trials. It showed about 35 times more heterozygosity than the common vetch genome and worked much better for alignment of long-read sequences from hairy vetch. So using that resource, Neil Toulouse, who's a research associate with Drs. Ridey and Kisin-Kusik, started work to get at the weediness of vetch, so hard seed and pod shatter. In other legume crops, both of those traits have been found to associate with one or a few large effect loci, which then lend themselves to marker-assisted selection. So they thought, let's see if we can find that in hairy vetch. They genotyped about 1,000 vetch plants from 80 common maternal parents, grew those plants in 2 environments, and they found a 68% reliability related to heritability for seed dormancy, which again is great news to breeders. We can potentially do something about the hard seed percentage in vetch. Neil found a large effect loci on chromosome 1 and is proceeding to, or they have developed a high throughput PCR phenotyping system to genotype, young germplasm. Our first soft seed nurseries generated with this system will go in this fall, which is really exciting. A manual hard seed test takes about 30 minutes per sample, so this is a huge improvement in being able to detect germplasm that could be favorable for soft seed. We found that either 1 or 2 copies of this gene is effective, but no QTL were found for pod shatter. So the next step was to look at different vetch germplasm pools and hope to both validate that QTL and maybe find some QTL that were present with lower allele frequency for pod shatter. So they used pooled phenotypically contrasting DNA samples from 4 site years not used in that previous study. And they did find 6 QTL for pod dehiscence, or no, 4 QTL for pod dehiscence. Unfortunately, they worked in the wrong direction. So they made pods more prone to shatter instead of less. But as Neil said, turns out science is hard. They also found 6 QTL for seed dormancy, which they'll continue to explore. But notably, you don't see that, you see no peaks on chromosome 1. So as we know, these association studies are dependent on the germplasm used, the environments they're grown in, maybe even sequencing techniques or sequencing runs. What Neil and Heathcliff and Lisa are discovering, though, that is not published yet, they think that the major marker that they found before is associated with a haplotype that was present in either very low frequency or not at all in these populations, and that it still is important for soft seed. The closest marker that with fairly high significance in this study did show that it was being selected down over years in our breeding program through those manual slow tests. So hopefully we have honed in on an important region, although we certainly haven't told the whole story with that one large effect locus. And finally, a bonus of starting to investigate the hairy vetch genome was discovering this outgroup. PCA revealed that there were 2 major subpopulations in our breeding program. The germplasm in this outgroup is derived from just a few parents, including purple bounty, which is a widely sown vetch cultivar that has been called hairy vetch. But these individuals are pretty distantly related to the Bahiya villosa or hairy vetch genome, and they also appear not to cross-pollinate with this core population. We consulted a legume expert at the University of Miami, Ohio, herbarium, and he gave us a few possible species that this could belong to, but didn't make a definitive ID. We're calling it smooth vetch just because it's smooth. It has much less pubescence. It's generally more determinate, earlier to flower. So it has some beneficial traits, but it's not hairy vetch, and it's not crossing with our hairy vetch. So we're taking it out of our breeding populations. We feel like this is a great example of what happens when you start to leverage genomic tools for these species that just haven't received much attention. There's a lot we can discover that has just not been on our radar before. Okay, shifting gears to a self-pollinated legume, winter pea. Winter peas are not quite as winter hardy as winter vetch, parentheses yet. but they do make a nitrogen contribution, and they're reliably soft-seeded. So there's no weediness issue with pea. Thus far, the CCB pea breeding program has worked primarily with Rebecca McGee, who's the USDA dry pulse breeder out in Pullman, Washington. And she's provided crosses that show promise, or that she feels are promising for the traits we care about. The CCB breeding program, most of the work I'll be talking about has been led by Elise Bastos Martins, now of Corteva, when she was a grad student at NCSU. And then it's recently been handed over to me. So these segregating crosses that we've received from Rebecca are planted in both research nurseries and on-farm nurseries. In some cases, winter does all of the selection. We just keep everything that survives. When we don't have much winter kill, we select on those important traits, vigor, biomass. Again, we make selections among them within families. The best lines are increased and advanced to our advanced line trials. Our advanced line testing network has allowed us to identify regions where pea survival is low but not zero, and then to strategically use those areas for selection. So that green circle up there is where winter survival is less than 50% but not zero. So that's where we're concentrating our on-farm nurseries, so we can leverage both those cold environments and those farmers, many of whom are farmer breeders, for their eye, their experienced eye. And a couple of selections that I'll talk about in a moment came from those on-farm nurseries. So the best performing lines we have so far are those from the participatory breeding program. So this analysis was also done for Albert Lee seeds. So it involves, it includes sites in the Midwest and Northeast. Wyo Winter was the best named check overall. So that's set at 100%, and everything else is compared to Wyo Winter. You'll see this trade-off. Can I figure out how to use this? Yeah, there we go. So between lower fall vigor and then higher winter survival biomass spring vigor. That's a common trade-off in plants, right? So plants keep their growth small before the cold season, which allows them to concentrate metabolites and do all those winter hardiness things, and then they really grow the next year. But we're not seeing that trade-off in these top two lines. So they're showing really nice fall vigor, and they're still surviving the winter, showing good spring vigor and putting on nice biomass in the spring. So that's exciting. The first one is being picked up by Albert Lee Seeds. The second one is, since it's a farmer bred line and this farmer runs a small seed company, he's going to be the primary licensee on the PVP, and he'll either do all the seed distribution himself or maybe sub-license to another seed company. So this is one of the ways that we're trying to place improved germplasm at multiple points in the seed system. Our network allows trialing in many environments, but there are only a few of our locations that are cold enough to really test winter survival. So to increase our insights into winter survival of these peas and to introduce farmers to our lines and the idea of growing peas as an overwinter cover crop in general, we're planning small-scale on-farm trials with about 60 growers this fall. We're going to use seed-linked trialing software, which is a participatory variety testing software, and we'll supply each grower with 3 or 4 lines, a mix of commercial lines and CCB developed lines. These will be small plots, like about the size of those advanced line trials. So a good fit for vegetable growers, even gardeners with substantial plots, people with single row planters, or the patience to plant by hand are who we're looking for. And we're excited to both expand our network of growers, especially to that smaller grower crowd. Many of the growers that we've been working with cover cropping, to get to 40 million acres, you need people who are operating a lot of acres. But cover crops have a really important place in smaller scale farming as well. And this is one of the ways that we can start making those connections and learning whether smaller farmers have different needs than larger farmers. Do we need to be breeding for different objectives, or is it more an education and extension game about getting people familiar with these practices? And finally, we are seeking new germplasm that's better adapted for both low temperatures and freeze thaw, or acclimation and deacclimation to cold. Steve Mulkey from the University of Minnesota has led the work on this front using growth chambers. To screen for freezing tolerance, he subjects these horse pea seedlings to a cycle of cold, then sub-zero acclimation, then freeze down to -12 Celsius, and then a recovery, and scores damage on a scale of 0 to 5, from no damage to complete death. He started a breeding program with the most freeze-tolerant accessions and ran field tests through Minnesota winters, and found a pretty impressive range of variation, from 0 survival to almost 100%. He selected the lines with more than 60% survival, then selected further on seed coat characteristics. And so these F34 lines here are currently planted in his nursery at Minnesota. Just was corresponding by email with him, and he said that about 5% of them are surviving. They had a very low snow winter, which is and very cold. So these are great selection conditions. And then we have some of the more promising F2 derived F3 families in our nursery here and on those on-farm nurseries in cold zones. So we'll see what happens. Finally, I was really impressed with this figure, which compares these F3 families with the F3 derived F4 families. So these are parents and progeny. In the F3s, we're seeing very few individuals per family, 0 to maybe a few, with no damage after this freezing protocol. In the next generation, we're seeing up to a third of individuals within families seeing no damage after the freeze-thaw protocol, and many more with intermediate scores. You can see the numeric scores, remembering that low scores are better in this case. So this is really exciting progress, and we're excited to keep working toward this, especially as freeze-thaw becomes more prevalent across more locations. We're seeing it, we saw it in Corvallis, Oregon this year. The Mid-Atlantic is seeing it more. As weather becomes more volatile, this will be a more important trait for us to consider. And last, but definitely not least, is cereal rye, which is the most widely grown cover crop for its ability to tolerate late sowing. When nothing else will grow in the fall, cereal rye will probably grow. Its cold hardiness and its abundant biomass production in the spring. The CCB rye breeding program began in earnest last season with the dual goals of breeding specifically for cover crop use, especially for northern locations, and also to leverage our North Carolina State's team ongoing work on allelopathy and rye. At Cornell and at the University of Minnesota, we started evaluating PI lines that showed promise for cold locations. And at NC State, they started work many years ago to identify rye lines with high allelopathy. So this rye allelopathy. Rye contains compounds, benzoxanoids, that inhibit the germination and growth of small seeds. They occur in both roots and shoots. Chris Reber-Corton started work about 20 years ago to identify genotype specificity of these compounds. And then Demek, Rebong, and Aliawaris have been working with protocols for, the goal of characterizing allelopathy in rye was to hopefully develop a high allelopathy rye that could be used strategically for weed suppression. And as a former vegetable grower who wanted small seeds to grow after my rye cover crop, I know there's a place for a low allelopathy rye as well. So the NC State team developed an in vitro assay to gauge the effect of rye on an indicator crop, and also a field assay using lettuce, which is small seeded, vulnerable to allelopathy damage, and grows well in North Carolina springs, which is when rye goes to boot, or to starting to go to head. You will notice that the allelopathy from these rye plants can either form a gradient, like so, or a ring, dramatically termed a halo of death. So once we identify lines with either high or low allelopathy, so the lines in yellow here have high allelopathy or a high level of weed control or surrogate weed control, and then a few lines with low weed control in blue. The next step is to integrate that into elite germplasm. So the NC State team has 3 advanced lines that will start nationwide testing this fall. They also made crosses between these high allelopathy lines and 2 winter hardy varieties. These are hot by northern crosses that will feed into a couple of other experiments. So these will, this is a Cornell-based program to select rye for improved cold soil germination and vigor, with the goal of developing varieties that will perform better in those late season tight planting windows after cash crop harvest in the northern region. PhD student Raksha Thapa is planting these lines both in northern New York and in this thermal gradient table, so she can select individuals that emerge just above freezing. And finally, the hot by northern crosses are planted in our nursery here at New York and also in Minnesota, in addition to the selections we made last year. We will make our selections this year, harvest the seed, send some to North Carolina so they can do allelopathy testing, which will then inform our selections from nurseries next year. So we can start diverging our group into a high allelopathy and low allelopathy group. And an exciting recent development in our rye program is that Lisa Kissing-Kusik, who's a Cornell grad, and one of the powerhouses behind the CCB Vetch program, is starting a new position as a cover crop forage breeder, focusing on rye, still in Wisconsin. She'll be taking the lead on the CCB's rye breeding program and starting with identifying rye idiotypes that will serve different production systems. So what combination of early and late maturity, high and low allelopathy, plant stature, et cetera, will serve cropping systems in different regions of the country and different crops. Okay, so cover crop breeding progress. We have new variety releases coming in vetch and pea. We have a marker for a soft seed. We are breeding rye toward high and low allelopathy. We're working on cold soil emergence and vigor in both pea. Well, we're working on that in rye and cold tolerance in pea. Frontiers, looking like fleshing out the back half of our breeding pipeline. So the steps around increase, commercialization, farmer adoption. We are working on expanding seed production regions and continuing to grow our connections with seed companies to add that logistical and financial sustainability to cover crop seed systems. Remote phenotyping will be really helpful to build rich, meaningful, and less labor-intensive data sets. We can breed more specifically for intercropping. We can look at employing genomic selection strategically in ways that it makes sense as genomic tools come along for the crops we are working on. So the path to 40 million acres is paved with, or perhaps sown with, continued education, networking, and creativity among growers and researchers. The relationship that facilitates all that and continues building this cover crop seed system with diverse points of entry and broad availability of high quality cover crop seed, and variety improvement. So traditional, unglamorous, yet effective field breeding and multi-site trialing, genomic analysis, controlled environment work for specific traits. If you are interested and curious about this, we're having a couple of field walks coming up on April 23rd and May 1st. You can snap this page or go to that QR code. That's our website on our events page. There's more information. Please come walk and talk with us if you're interested. We'd love to show you what's out there. Thanks to the Moore Lab, without their hands-on work in the field and handling seed, this would not be possible. And thanks to the CCB Network team and our funders. Grateful for the chance to talk with you all. Yeah.
This is about the work your collaborator in Minnesota was doing.
Mm-hmm.
How are we, with the field trials for the freeze-thaw tolerance and winter pea, how are they distinguishing that phenotype from just like winter survival scores?
So you mean the controlled environment phenotype with the field?
When they're doing the field trials for freeze-thaw now, how are they just going at the end of the season in the spring and scoring winter survival broadly, or are they getting more data points along the season?
I think the controlled environment trial is to select germplasm for intermating or to select what goes into the field. And then whatever conditions happen that season, select for whatever survives.
Okay.
but it's you know, it's hit or miss because you don't know what you're going to get. Yeah. Good question. That's a great question. And Denny, you're welcome to weigh in as well. I think there is a point at which too much is too much in terms of, say, biomass, depending on the cropping system, depending on how they plan to terminate it, in terms of whether we get to good enough and then move on. Yeah.
Like whether, or one of the things I think about is like whether the cover crop market as it currently is can support, you know, an infinite number of varieties, a many number of varieties.
Yeah.
I don't think we need 200 different varieties.
Right. But
for cover crop purposes, right?
Mhm.
And as the industry.
Yeah. Yeah, exactly. I think maybe that radiation happens with like filling more niches. And, you know, say for vegetable growers, which especially like smaller scale diversified vegetable growers like I was, there could be lines bred for those planting and termination windows that are offered by seed companies that really target that market, that are small or mid-size and maybe very regionally adapted. That makes sense to me, including some of these workhorse broadly adapted varieties that are really widely available.
Especially as we're starting.
Yes. Exactly.
We can't read just for Northern Iowa and versus other, you know, like
right.
Our target population and target management system.
Right. And rye is really an easier nut to crack because it's so widely used and it's often used in monoculture. So the testing is much less complex, whereas legumes are often grown in mixtures with various small grains and are just less widely used than rye by a long shot. So it's both initial adoption of try this species at all, and then dialing in what traits within that species are going to optimize the system. Yeah. Yeah.
Great work. I was wondering regarding the rye. Is that a trait that seems to be like something highly seems to be throughout the entire life cycle of rye?
Oh.
If not, is there anything or what really things that are highly compatible?
I mean, that sounds great. We're only really at the beginning of investigating this trait. So what we know now is allelopathy in, I believe, all tissues of the plant is greatest at boot stage, or when the plant starts to make a head, right? And the head emerges from the leaf sheath. In terms of distribution between root and shoot, in terms of, you know, some of the lines that have been identified as high allelopathy, some of our discussion within our group has been, are these lines just earlier? You know, are they just phenologically more advanced? And that's why they're producing more of this compound. We're also seeing, we have some preliminary results in a greenhouse screen where they grew, I think, 4 different rye lines with known different levels of allelopathy, harvested the shoots, and then planted various species of crops or weeds, and saw very different effects on different crops, and sometimes like rye line by crop interaction. So this is very complex. And so, yeah, we're trying to, well, this is one of Lisa's jobs, is to figure out how do we start to parse out these different variables to figure out how we strategically use this trait? Yeah, it's cool and powerful to just like blast the weeds out of a field, but if you get allelopathy that strong, like there is evidence that it also affects large seeds like corn and soybeans, and that's not what we want. So yeah, it's an interesting one. Yes.
So you briefly mentioned are there measurements to be taken on throughout the program, how much nitrogen is becoming available and whether it's available for how it's thrown afterwards?
Not right now. I know there was biological nitrogen fixation data taken several years ago, and that's how we came to that conclusion that nitrogen fixation is so highly correlated with biomass. Let's measure biomass because it's so much easier. But in terms of release over the plant's life cycle, vis-a-vis availability to the next plant, I don't believe that's been done in like a time sequence. Yeah, okay. Right. I believe that most of them are for employing the practice. So you sign up for, I'm going to put cover crops on 200 acres. Somebody from the soil conservation district comes out and verifies, yes, you've done that. You get your incentive. I think some of the private programs are starting to try to quantify change over time.
They say yes or no, you're doing the practice.
Yeah.
There's also only that payments for a lot of programs, like if it's planted before a certain date.
Yes.
After data is kind of a practice that is actually having a benefit. You're not just not
right. But that, yeah. Yes.
Yeah, and that's part of the exciting part about our collaboration that because their guidance about cultivar, like cultivar adapted regions are then feed into the like what the
recommendation tools. Yeah.
So,
yep.
So yeah, they have a
and there's also some programs are designed for new adopters of cover cropping. Some will incentivize people or give them benefits for what they're already doing. Yeah, it's very diverse and changes every year with policy. Yeah. Yeah. Well, this would be Ginny's wheelhouse because she's been at this for more years than me. I just came on last year, so.
From the Department of Plant and Agroecosystem Sciences at the University of Wisconsin-Madison. Sorry, I'm very bad in pronouncing these names. Under the mentorship of Dr. E. Wang. And then her research project focuses on development of remote sensing-based decision support models for nitrogen management of vegetable crops such as dark kidney beans and snap beans. And today she'll be presenting some of her work in the talk titled Eye in the Sky, Hyperspectral Imaging for Sustainable Nitrogen Management in Vegetables. I would like to welcome Taqdeer, and yeah, you already have them.
So our team is working with kidney beans and snap beans. Both are vegetable crops, and past research shows that has shown that excessive application of nitrogenous fertilizers could lead to nitrate leaching and groundwater contamination. Excessive application of nitrogenous fertilizers also leads to increase in the input cost. The graph on this slide represents the change in the prices of fertilizers over past few years. And we could see that after the year 2020, there was substantial increase in the prices of nitrogenous fertilizers. This map represents the groundwater contamination due to nitrates in Wisconsin. And studies have shown that 20 to 30% of private wells have nitrate level that exceeds 10 milligram per liter. Excessive application of nitrogen fertilizers also increase the disease pressure, such as white mold in case of kidney beans and snap beans. And higher disease pressure reduces the crop yield. Previous study conducted on dry beans suggested that there was higher white mold incidence when higher nitrogen rates, such as 179 kg per hectare, was applied. As we know that every human being has slightly different fingerprints, and it is possible to identify specific human being those fingerprints belong to. And we could do similar thing with plants, even though they don't have fingers. Well, the fingers I am talking here do not, the fingerprints I'm talking here do not start with fingers, but with light. When light fall on plants, some of it is absorbed by the plant, but certain portion of the light is reflected back into the atmosphere. And the hyperspectral sensors installed on airplanes or drones or satellites capture the light reflected back by the plants and provide us with the information that some of it we couldn't see with the naked eye, also known as spectral signature. The graph on this slide shows the spectral signature of a healthy plant shown by darker green line is different from spectral signature of a nutritionally deficient plant. Crop growth is largely affected by the interaction of genetic, environment, and management factors. And we are using hyperspectral remote sensing to understand the spatial variability within and across the crop fields. The overall objective of our research project is to use aerial hyperspectral remote sensing for understanding how snap beans and kidney beans respond to nitrogen fertilization on irrigated sandy soil. During the first half of the talk, I will be talking about snap beans, and during the later part of the talk, I will be covering kidney beans. This is the field design of snap beans, where we applied 6 different nitrogen treatments shown by different colored rectangles on the RGB image. And we applied nitrogen treatments at 3 different growth stages to 2 different cultivars, one of which, DM88, was a nodulating cultivar, and second was a non-nodulating cultivar. This is a timeline of in-season nitrogen application shown by 2 blue bars, hyperspectral imagery collection shown by orange bars, and whole leaf sample collection shown by 2 green bars. The red rectangles on this slide represent the combination of hyperspectral images and whole leaves that were used for in-season nitrogen prediction. And all the hyperspectral images were used for end-of-the-season crop yield prediction. We collected hyperspectral images using high-spec sensor by installing it on airplane, and we collected images on weekly basis. After extracting the reflectance values by drawing the polygons using QGIS software, we selected the top 20 wavelengths that highly correlate with the end-of-the-season crop yield and in-season nitrogen status of the plant. And we trained different machine learning models by using top 20 wavelengths along with genetic, environment, and management factors. The genetic factors were cultivar, environmental factors were soil temperature, precipitation, growing degree days, and management factors were nitrogen rate, irrigation, and days after planting. This slide shows the crop yield of 2 different cultivars of snap beans. We observed that non-nodulating cultivar, that was Huntington, was having significantly higher crop yield when nitrogen was applied at 112 kg per hectare or higher than that. However, DM88, which is a nodulating cultivar, was having significantly higher crop yield at 84 kg nitrogen per hectare. That means that DM88 might be fixing nitrogen, and it needs less application of nitrogen fertilizer. This graph represents the leaf nitrogen percentage at different growth stages of snap bean plants. We observed that higher nitrogen rates were having higher leaf nitrogen percentage, and overall leaf nitrogen percentage decreased with increase in the growth of plants. This slide shows the spectral signature of snap bean plants at 6 different growth stages. Each graph has 6 different spectral signature curves representing each nitrogen treatment, ranging from 22 kg nitrogen per hectare to 168 kg nitrogen per hectare. And the x-axis of these graphs is wavelength in nanometer, which is further subdivided into visible, near infrared, and shortwave infrared region. The y-axis is reflectance values. The red bars in these graphs represent the top 20 wavelengths highly correlating with the crop yield. We observed that this pattern of spectral signature changed across the growing season. Since in the beginning of the season, the reflectance values in the near infrared region were not that high. However, they increased substantially later in the season. Also, different nitrogen treatments were having specific spectral signatures. The lower nitrogen treatments shown by yellow-colored lines were having higher reflectance values in the visible and shortwave infrared region. However, the higher nitrogen treatments shown by darker green lines were having higher reflectance values in the near infrared region. The red bars that correlating with the crop yield also changed at different growth stages. They were in the shortwave infrared region in the beginning of the season, and they shifted to the near infrared and visible part of the spectra in the middle of the season, and they shifted more into the visible part of the spectra later in the season. This table represents the results of random forest model, which outperformed as compared to other machine learning models at predicting end of the season crop yield when it was trained using the combinations shown in the first column. Random forest model performed best when it was trained using top 20 wavelengths along with genetic, environment, and management factors together as compared to all the other combinations. And our model performed really well starting from the V4 to V5 growth stage, meaning that we could use the imagery for crop yield prediction very early in the season. These two images were used for in-season nitrogen prediction, and the red bars in these images represent the top 20 wavelengths highly correlating with the in-season nitrogen percentage during the season. And we observed that the top 20 wavelengths were in the visible and near infrared region at 42 days after planting, and they slightly shifted to the right side of the spectra. Again, we observed that random forest model performed better as compared to other models at predicting in-season nitrogen percentage when it was trained using top 20 wavelengths along with genetic, environment, and management factors. And the model performed better at 42 days after planting as compared to 50 days after planting. This is a field design of kidney beans where we applied 8 different nitrogen treatments to 3 different cultivars at 3 different growth stages. We observed that the crop yield response was very much cultivar specific since there was no significant difference in the crop yield of cultivar Dynasty. However, the crop yield of Red Rover and Montcalm were significantly higher at higher nitrogen treatments. These 3 graphs represent the leaf nitrogen percentage across 3 different cultivars, and we observed the similar trend that higher nitrogen rates were having higher leaf nitrogen percentage, and the overall leaf nitrogen percentage decreased with the growth of plants. We collected 9 different hyperspectral images at different growth stages of kidney beans, and we observed that in the beginning of the season and at the very end of the season when the crop is completely dry, the spectral signature or the reflectance values in the near infrared region were not that high. However, in the middle of the season, there was substantial increase in the reflectance values in the red edge region. Also, similar to snap beans, different nitrogen treatments were having specific spectral signature, and that was more evident in the middle of the season. We didn't really observe any specific pattern of the top 20 wavelengths as we observed in snap beans, but we did observe that at certain growth stages, they were more scattered, and at certain growth stages, they were more clustered. For kidney bean yield prediction, linear regression model performed better as compared to other models, and it performed best when it was trained using top 20 wavelengths along with genetic, environment, and management factors together. But overall, our prediction for crop yield of kidney beans was not as good as snap beans. These 2 dates were used for in-season nitrogen prediction of kidney beans, and we observed that the top 20 wavelengths highly correlating with the leaf nitrogen percentage were in the visible part of the spectra, and they were more clustered. And at 50 days after planting, the top 20 wavelengths were more scattered. And random forest model performed better at predicting in-season leaf nitrogen percentage of kidney beans when we trained the model using top 20 wavelengths along with genetic environment and management factors together. In conclusion, we observed that higher nitrogen treatments lead to higher crop yield and leaf nitrogen percentage, but the response is very much variety dependent. And the spectral signature change across different growth stages, and different nitrogen treatments have specific spectral signature. The top 20 wavelengths also change across different growth stages, meaning that we might need different parts of the spectra for predicting crop yield or in-season nitrogen percentage at different growth stages. We could use machine learning models for kidney bean and snap bean yield prediction, but we need both GEM factors along with plant spectra. The future work will focus on validating our models in commercial field settings across different environmental conditions, soil types, and on different vegetable crops. And the long-term goal of this project is to develop whole field maps that will help vegetable growers optimize the in-season fertilizer management by keeping in mind a certain yield goal. I would like to thank all the lab members who helped in data collection and our collaborators who helped in hyperspectral imagery collection. I would also like to thank the Department of Agriculture, Trade, and Consumer Protection for funding this project. And huge thanks to Synapsis and the organization committee for giving me opportunity to speak here today. Thank you.
What is reflecting at 2 microns? What is it?
Sorry, can you?
So you're getting a signal at 2,000 nanometers. What's the source of that? What's bouncing off? What's coming back?
Sure. So the question is that the reflection at 2 micrometer, like what is leading to that reflection? So maybe I could go back quickly to the the slides here. But usually in the visible part of the spectra, the reflectance is due to chlorophyll absorption or absorption of different pigments. But in the near infrared region, the reflectance is due to plant structure or canopy structure or cell structure. And in the shortwave infrared region, the reflectance is due to the water content of the plants or dry matter accumulation. Does that answer your question? So the question is that what is our hypothesis that early point is more predictive for crop yield prediction? So we observed that like the R-square for crop yield prediction was really good in the beginning of the season, and it could help. Like for example, at 25 days after planting, we haven't applied our second nitrogen treatment yet, and if we could, we would know that how much crop yield we would get, and if we would be able to find that how much nitrogen crop needs or how much nitrogen is there in the plants right now, it could help in determining the fertilizer management or like making the management decisions. So for kidney beans, we didn't really get like really good prediction early in the season, but for snap beans, we did get really good prediction at early growth stages. And the reason behind that could be that like, and that the top 20 wavelengths were in the shortwave infrared region. And that is the region where most of the nitrogen absorption bands are. So, and we had already applied the first nitrogen treatment by that time. So that might be reflecting due to the first nitrogen treatment application that we are seeing differences in the plants, and we are able to see like what the crop yield would be early in the season. Yes, we did. We saw white mold on kidney beans, but not on snap beans, because that was a very short duration crop. And higher nitrogen treatments were having, like, although I haven't included the data here, but higher nitrogen treatments were having significantly higher white mold, especially later, like closer to the harvesting. Yes, there were, like, although from our results, higher nitrogen treatments were having higher crop yield, but that white mold could be a confounding factor, but we don't really know that how to take that out for measuring yield. Any other question? Yes. Yes, we're actually trying different techniques. Like, we're also trying 5 bands or the whole spectra or just 1 band. And we are also trying PCA to see like which bands, but like we were like, when we were choosing 5 bands or 20 bands, we were not getting too much different results. So we are working on that to reduce the number of bands so we could use take more advantage of hyperspectral imagery, and we could use this data for maybe designing a multispectral sensor that, or like deciding that which region or which wavelengths are most important at a certain growth stage. Yes.
Do you think that specific metabolites?
Yeah, we do, like, the spectral signature is changing actually due to like due to the changes in the metabolites in the plants. And for example, later in the season for kidney bean spectral signature, the plants are dying, and that's why we are seeing slightly higher reflectance values in the red region, which might be due to carotenoids, but we haven't really done any kind of chemical analysis yet to see where the absorption bands are, or like what is exactly leading to the change. Actually, when I started the project, I started with calculating different vegetation indices, and we calculated 26 different vegetation indices, but we were not seeing a particular trend. For example, they were like at certain growth stages, one vegetation index was doing really well, but at certain growth stage, there was another vegetation index which was doing well. We were not able to generalize, and then we were having so many wavelengths, and we thought, why not like play around with data and see what the like better information we could get out of it, and decide that what wavelengths will be more helpful. So that's a really good question. That is, the irrigated sandy soil is in central region of Wisconsin, where most of the snap bean growers are, and they grow beans, or even the kidney bean growers are. And we like there is a huge nitrate leaching problem there. And another objective of this project was also to optimize, or to like find which nitrogen treatment could be best for getting higher crop yield, as well as it is best for from environmental point of view. So that was the reason we decided to choose that region. But in Wisconsin, there are another soil types as well. In northern region, it's not completely sandy soil. It's more of a clay soil or silty loam soil.
Well, it is my honor to introduce Dr. Nolan Bornowski. Nolan is a research scientist at the mushroom spawn company, Amycel Spawn Mate, where he focuses on improving the white button mushroom through genomics and bioinformatic approaches. Before working as a mushroom breeder, he got his master's from Michigan State in the dry bean breeding and genetics program, and then obtained his PhD from MSU under the guidance of Dr. Robin Buell. Today, he will present some of highlights in mushroom breeding with his talk titled Out of the Darkness, Historical Challenges and Modern Solutions in Button Mushroom Breeding. Please welcome Nolan to the stage.
Okay, great. Thanks everyone for coming out. Had some great talks. So yeah, today I'm going to be talking about a very unusual crop from a plant perspective. We grow button mushrooms at our company here. And it certainly has a lot of complex challenges associated with this crop. So here's a little bit of an outline. I'm going to start with an introduction to introduce you to myself, my company, and a little bit about the system that we work with. Then I'm going to dive into some of our breeding challenges and how we kind of approach them. Finally, I'm going to conclude with some of our research highlights, and a future outlook on the industry and our company. So a little bit about me. I like to describe myself as a scientist who works with DNA sequence to make better plants. And for the past few years, I can also include fungi in that. This crazy mushroom looking guy was actually me from a few years ago. I'm still the crazy mushroom guy. It's just harder to tell now. So anyway, I have a background in plant breeding. I've worked with different plant species. I worked in potatoes as an undergraduate researcher. And then for my graduate degrees, I worked in traditional breeding with the dry bean breeding program. And then I pursued my PhD in more of like a computational and genomics area, where I worked in maize and also some orphan crops as well. I had a stint in the popcorn breeding sector, which was also very fun. We got a lot of free popcorn samples there. And currently, I'm a research scientist where I use genomics and bioinformatics to help make better mushrooms at the spawn company, Amycel Spawn Mate. So you may be wondering, who is Amycel Spawn Mate? Well, we're kind of a big deal in the mushroom industry, and it's okay if you haven't heard of us. We're actually one of the 3 major spawn producers in the United States. And so how the system works is spawn is what we use as seeds to plant our crop to grow mushrooms. And the spawn is just actually grain that's colonized by mycelia. So our 2 main goals are to develop new mushroom strains. And we also make spawn for our mushroom growers. Our company is vertically integrated. This means that we can grow mushrooms on our own farms, and we have farms across the United States, Mexico, and France, so we can bring our consumers very fresh product. We can move these mushrooms around using a transportation company that we are also associated with. And then we sell our mushrooms under the Monterey Mushrooms label, if you ever see this in the grocery stores. And our main focus, and what I'm going to spend most of the talk today on, is Agaricus bisporus, or the button mushroom. For reference, we have about 75% of the brown mushroom market share. And the brown mushrooms in this context are the same as the white mushrooms. They're all from the same species, Agaricus bisporus, just with a different cap color. So, like I said, we're one of the major spawn producers in the US. Our office is located in a very modest building in Central Coast of California. And in addition to those major goals there, we also serve as a research engine. So there's a lot of unresolved science and questions in the mushroom breeding realm. And we have a small group, but we also spend a part of our resources to develop resources to try to better understand what are we actually doing and how does our crop work. So it's a very small group. We have a director. We have about 5 research scientists on staff. We have lab assistants to help with the wet lab things, support staff, and some growing technicians who actually are in charge of spawning or seeding the rooms and creating our trials and then harvesting the mushrooms. We also provide other services to our company. We certify our strains before we release them commercially. We test and analyze mushroom substrates. And this is important. This would be analogous to doing soil testing services to bring it into a plant realm. We develop new products, and we also provide strain ID and disease testing for some of our companies that we sell our spawn to. We sell the seed. So the button mushroom, we've kind of already established it's a pretty niche crop. It's a hybrid crop that's colonially propagated, and it's perishable, but it has high value and requires high input. So just looking at that, I think the closest plant system that I can think of, it's pretty similar to strawberries in this regard, but certainly other berries could be considered as being similar too. I pulled some data from the 2023 US economics to look at the crop values. And I have them sorted according to this leftmost y-axis. So the data will be highlighted in blue is the total production value in billions of dollars. And obviously, corn is king here, no surprise there. And soybeans are also take up a lot of the market. Mushrooms are down over here. They have a very small total value in terms of the economic output in the US. However, I would like to highlight the right side y-axis, which is the price per pound. So you can see that they have very high value in this regard. Also similar to strawberries and some other, you could say, more minor crops. I'm going to focus and highlight this little modest segment here. We have about $1 billion of total value in 2023. And this pie chart is breaking it down by the different sectors. And the blue is what I would like to highlight. Most of our mushrooms go towards fresh market. However, we do have mushroom segments that are used for processing, like would go in like a processed soup, for example. And there's also a segment for specialty mushrooms. These would be other mushrooms like you may have heard of, like shiitake or oyster or turkey tail, things like that. However, most of our revenue stream comes from breeding agaricus bisporus for fresh market. So I need to introduce a little bit about button mushroom biology. They belong to some fungi called the basidiomycetes. And they're called club fungi because of the shape of their spores. They kind of look like a club. The white mushrooms and the brown mushrooms are all part of this club, if you will. So the only difference is they have different cap colors. Furthermore, the differences between what you might see in the store market as cremini mushrooms or baby bellas and portobellas is just a difference when they were harvested. They were harvested at different maturities. There are certain considerations from a breeding perspective if we want to breed for cremini mushrooms, which are harvested small, versus portobellas, which are harvested later, considering the speed that the caps develop and the gills open up. So some other basidiomycetes you may have heard of include polypores. These are like the shelf fungi that grow on trees. An example of this here is turkey tail. It's beautiful. And from a plant scientist perspective, I had grown so accustomed to thinking of fungi as the enemy because we typically see them, in this kind of pathogenic world. So some example of other basidiomycetes include rusts and smuts. As far as when I say the term mushroom, let's just get on the same page. The mushroom is just the fleshy, spore-bearing fruiting body. And to make analogous to plants, we have our mycelia down here, which would forage for nutrients like the roots, the stem, which provides support to the structure, and then the marketable portion up here is the cap, and this would be the fruit or the part of the plant that's sold. Now I'm going to introduce a few concepts that will be most likely foreign. So they all come from Greek, which is really fun if you're a fan of my big fat Greek wedding. But these terms are homokaryons versus heterokaryons. So in this fungus, we have homokaryons that are either spores or mycelium that have an identical nuclei type. And these would be our haploids. We also have heterokaryons, and this refers to spores or mycelium with two different nuclei. These are also called dikaryons because they have one haploid set coexisting in the same cell with a different haploid set. I found this life cycle figure from a textbook, and I think it demonstrates a general basidiomycete life cycle. There are some quirks about our specific mushroom that we're talking about here, and I will be sure to describe them. So let's start at the beginning, as they say. In step one here, we see mycelia of two different mating types. colored as a blue mating type here and a red mating type here. These mating types, if they're compatible, will fuse together in step 2 here. The step is called plasmogamy, and they'll produce this dikaryotic mycelium. So again, these 2 nuclear types are coexisting in the same house or in the same cell. Then step 3. Step 3 occurs when we provide the correct substrate and the right environmental conditions. The mycelia will grow and produce a fruiting body. This is also called a basidiocarp. Inside of the gills of this basidiocarp, we have our 2 nuclei still coexisting. They haven't necessarily combined yet. They're still existing separately. They're still existing separately inside the basidia. So now we're getting into starting the reproductive process, starting to create the spores themselves. In the basidia, they undergo karyogamy. So the 2 nuclei finally merge together and then into one cell. And this is where we get the diploid zygote portion of the life cycle. This undergoes meiosis. It's not a typical meiosis. There's some quirks about that, which I'll talk about in future slides. But as a general result, we do expect to see 4 haploid nuclei as the result of meiosis. These occur in the basidium. They're not yet packaged into spores. This is where it gets a little bit weird with our crop, because in typical basidiomycetes, they have these 4 spores, and each spore gets 1 haploid nucleus. However, in Agaricus bisporus, there's typically only 2 spores produced, and each of these spores contain the parental nuclei. So essentially, it's repackaging its meiotic products into a spore that can be used to recreate itself. And this is a challenge. So finally, in step 8, we get this dispersal and germination. In the very rare case in our crop, we have basidiospores containing 1 haploid nucleus, and these would be considered, these homokaryons will germinate to create haploid mycelia. This happens very rarely, unfortunately. The more common case is where we get dikaryotic spores germinating to produce dikaryotic mycelia that in turn will create dikaryotic mushrooms without having any kind of introduction of genetic diversity. So the button mushroom requires a nutrient. They require certain substrates to grow. And in nature, they get these nutrients from manure or leaf litter. Shown here is an example of a cypress tree. And these are native to the central coast of California, where my company is located. And button mushrooms are found commonly associated in the leaf litter of these cypress trees. However, from a production standpoint, we create our own substrates. In the case of spawn, this is the grain that's colonized by our hybrid mycelia. For agronomic trials, we use small amounts of spawn. We put the grain in flasks and inoculate the flasks with little pieces of mycelia. These mycelia then colonize the grain until you get this mixture here, which is ready to plant, or we call it spawning. In a commercial setting, we use these larger bags filled with grain and nutrients and mycelia. These bags have strips that allow the exchange of heat and gases, and they're stored in these climate-controlled rooms so we can better monitor their growth and progress. We also generate our own compost. This is done at a very large scale. You can see here, this blue circle is highlighting a person for some scale. This compost is generated from mixing wheat, horse manure, oil, and some nitrogen, and we let it naturally break down for a couple weeks. It also undergoes a pasteurization step after this, which breaks it down even further. Now, this is helpful because this changes the microbiome of the compost. And in general, Agaricus bisporus is pretty tolerant of outcompeting any other kind of microbes in the compost, so it doesn't have to be like sterile, if you will. When we get to actually cultivating the mushroom, we plant them. This is called spawning, where we mix our spawn and our compost. The mycelia will colonize this compost in all directions. And after about 2 weeks, the compost has been colonized enough, and we add a casing layer. This is just a low nutrient layer that the mycelia will grow into, they will expand, and it will force the mycelia up into this layer. This also helps with watering. So just like any other crop, we need to water the mushrooms, and making sure that we have enough airspace for the water to actually get used up by the mushroom is important. Next part is we flush the room, and this is also referred to as a break. This is where we stimulate fruiting by manipulating the environment. So we decrease the amount of carbon dioxide that we pump into the rooms, and we lower the temperatures. And this stimulates the mushroom to say, okay, it's time to create a mushroom. Typically, we harvest these mushrooms over 5 days or so. Then we allow them about 3 or so days to regrow and reform new mushrooms. In total, we repeat this process about 2 to 3 times. So we have 2 to 3 breaks per planting. And each time we can say we get about 4 to 8 pounds per square foot per flush. Obviously, there's yield decreases with each flush that we get. And this is typical, like you would see in like other ratoon crops, for example. In our case, it's due to disease pressure mostly, but also the nutrient availability will decrease slightly. So here's an example of where we grow the mushrooms. Contrary to popular belief, we don't have an elaborate cave system. We grow these mushrooms in the case of, for our commercial tests, in these walk-in freezer type rooms that are climate controlled. And when we do these tests, we grow them in smaller wooden boxes that are filled with compost and mycelia, and we fruit them with mushrooms. And this is, at this point, I need to mention that this is a year-round endeavor, the growing the mushrooms, because we have them in a climate-controlled environment. There's no planting season or growing season. Planting season is every day. Growing season is every day. In fact, we take measurements on these mushrooms every day because their growth is so fast. So we're always collecting field data every single day and using that to quickly turn over and generate our blinds that we want to select. And here I have a picture of our newest member to the team, Chloe. They were a former Cornell grad from the plant pathology department, so it's great to have them on board collecting this important agronomic data for us. On the right side here, I'm showing an example of the commercial production rooms. So this is done at a much larger scale, obviously, than how we would test or trial our new strains. And you see these are also very large wooden beds or trays that are filled with compost and spawn. And mushrooms are grown and manually harvested. So this requires quite a large amount of labor. Now I've kind of given you the broad overview of the steps in the button mushroom breeding production. I'm going to lay it out and put it all together. And this is from the perspective of a mushroom breeder. So we start with our germplasm, as all good breeding programs do. We have our proprietary mushroom strains. From these strains, we isolate spores. We let the mushrooms grow and mature, and then spores will drop, and we can pick out individual spores and isolate them. We grow these up on plates and let them germinate into mycelia. Crossing is actually pretty easy. You can just take a chunk of one mycelia and put it on a plate with another chunk from a different one. And if you're lucky, they will hybridize and grow together. So they will essentially merge and cross with itself, all by itself. Once we have this mycelia, we transfer this to a mixture of grain and nutrients. This would be our spawn. You can see a little more clearly in this example, a commercial spawn bag here with these breathing strips. From the spawn, we combine the spawn and compost, and under the correct management practices, we can grow our mushrooms. These mushrooms are then harvested and packaged, and finally they are shipped to the end user where you can go to the store and purchase them and put them on your pizza or whatever you want to do. From a breeder's perspective, this growing stage, this agronomic data is used where we select our mushrooms based on this stage. However, I've highlighted in these red outlines here, there's other considerations that we need to think of. There's other stakeholders involved in this process. We need to consider the growers. How do we make this crop grower friendly and resilient to different environmental conditions? But also the consumers, freshness and flavor and certain cap characteristics are also important. So I'm going to talk a little bit about the cultivation history. So when I talk about button mushroom breeding, it's a very recent phenomenon. And like any good former corn person, I have to mention maize in one of my presentations. And I just put this in here because this happened, maize domestication happened about 6,000 years ago. And I like to highlight this because you can see the different morphological changes that breeders change this crop from the wild grass teosinte to the corn that we work with, and grow on millions of acres around the world today. So our mushroom story, actually begins in the 1600s. This was the first time there was a concerted large-scale cultivation, and this was done in underground systems in France. In the 1900s, we see large-scale spawn production in the United States. So this is consciously generating spawn that can be used to grow mushrooms and introducing good practices to maintain single strains or single lines of spawn. The early 1980s saw a milestone development, and this was the development of the forced U1 strain. This was the first time that there was actual deliberate crossing or deliberate breeding going on in this crop. I have this gray arrow here because this U1 strain is the progenitor for most of the white button mushrooms that you will see even today. 2016 was a big year because there was a scientist at Penn State who developed a gene-edited mushroom with the goal of reducing cap bruising. However, since there's no actual published paper or research from this. All we get was this research highlight. And since then, there haven't been any commercial gene-edited or transgenic mushrooms on the market. Looking to the future, I think it's important to have an open mind with not only gene editing, but also where can we take the crop? I'm going to look back at maize and you can see these dramatic morphological differences. I would argue that right now, mushrooms are still in the domestication phase rather than the improvement phase. So I think I'm really excited to see where we can go from here. And as a genomicist or someone who works with sequence data a lot, I would be remiss if I didn't talk about reference genomes and the button mushroom genome. We had our first genome assembly was published in 2012, and currently we're on version 3. These are genomes for the constituent homokaryons of that U1 strain that I mentioned. And specifically, I'm going to give some metrics for H97. This was one of those haploids that were involved in that cross. There's about 31 million base pairs in this genome, which would take you a long time to count. But in more like general terms, it's similar with other fungi, but it's not really that big. We scaled it here 100 times, and you can see it's really quite small. compared to some other popular organisms that are well studied. And this is great for anyone who wants to do sequencing or genomics because we can sequence a lot of things and get great coverage. So the BOOSCO is just the number of single copy orthologs, and it's used to measure completeness of the genome assembly. And we're doing pretty good at 93% of those orthologs present in single copy. It has about 12,000 genes. For reference, it's about half as many genes in humans and about a third as many genes in maize. And we also have about 12% of the genome was found to be transposable elements. So that was some background. And now I'm going to get to the meat of all of the challenges that we're facing. I've kind of broadly separated them into breeding challenges and biological challenges, but there is going to be some overlap. The first challenge is there's a brief domestication history. We saw in the earlier slide, crossing really has only been going on for about 50 years in this crop. The good news is that today we have modern tools like sequencing and using good database management and experimental design. So this will allow us to catch up to other crops. There's also this narrow genetic base. This U1 strain has been around for the past 50 years and continues to be used in many commercial programs. So to introduce diversity, we can go back and use wild germplasm. We can also use molecular tools like create the diversity if we can't find it in nature. This would involve things like mutagenesis or gene editing. Again, I have to mention that no commercial mushrooms currently exist that are derived from those tools. There's been limited scientific research involved. Obviously, it's a niche crop, and there's only about a handful of research institutions in button mushrooms around the world. So we have expertise that is kind of spread thin. And as a solution, it would be great if we could increase funding, gain some more awareness, attract a different talent pool, and also collaborate with people who, we have a small group at our company, but if we can outsource some work or some questions to other groups, that would be great and help us make progress. Another consideration is it's difficult to protect our IP. So mushrooms kind of exist in this legal limbo. They're not animals, they're not plants. We do have the ability to use utility patents, so that's something, that's good. But in general, they can be clonally propagated. So what would be the incentive in spending all of these resources to develop a new strain that a competitor can just go and take without fear of legal ramifications? And so we would like to work more closely with regulatory bodies to kind of make sure that we can set a legal landscape that would help us create and protect our strains. On the biological side, I've hinted at this, it's difficult to produce our haploids. And we also have limited genetic recombination. To address this, we can use markers, we can introduce new germplasm, but this is one of the main challenges that's been facing the industry for quite some time. We also have mating types. So even if you do isolate haploids, the presence of mating types means that they may not be compatible. They may not actually fuse together if they have the same mating type. Again, we can examine to see if our germplasm is having this certain mating types using markers and sequencing. Mushrooms require specific growth conditions. It would be important to have mushrooms that are resilient, not only to different conditions and different management practices, but also have the ability to adapt to those different management practices, like how much water is being used and what are the temperature changes like in that growing environment. And last but not least, quality is important. So if you've ever gone to the grocery store and tried to purchase mushrooms, you know that by the time it gets to the end user, maybe they're not as fresh as you would hope. Well, we can address this by looking at consumer panels and breeding, making a conscious effort to breed for quality. I have gene editing here as a question mark because this was what that 2016 Penn State researcher was kind of thinking about, the quality aspect in reducing cap bruising. Okay, so I'm going to dive into challenge number one, was this narrow germplasm base. Remember when I said we start with these unique strains, this unique germplasm? Well, actually, it's kind of harder than it seems to have that unique strains. Before the 1980s, we had these heirloom strains where growers would grow a mushroom, they would collect these dikaryotic fertile spores and culture them up and grow a new mushroom from that. The problem is there's not really a lot of genetic recombination in the crop, but we do get some rearrangement of the parental chromosomes. So you're basically just shuffling chromosomes around these different mushroom strains. And then in the 1980s, we saw the development of this Horsey 1 strain, and it was a cross between two haploids from different traditional strains. Immediately, they saw yield and quality improvements compared to the existing strains, and it achieved rapid market dominance that continues to this day. To get new varieties from this, they developed, they took those dikaryotic fertile spores like this and grew up new mushrooms, much like this process that was being done previously. So there's still not a huge genetic base for our crop. Okay, so we say, okay, well, what if we solve this challenge? We solve the uniqueness challenge. Well, now we can just isolate spores, right? Well, the problem with that is that it's harder than it looks because most spores we can't use for breeding. We have a unique sporogenesis process that inhibits the formation of these haploids. So just to demonstrate this, here's our N plus N, our heterokaryon or dikaryotic sporocarp mushroom. In the gill tissue, we have, again, the dikaryotic basidium hanging out. This will fuse to create our diploid zygote and undergo meiosis. So in the post-meiotic basidium, we have our 4 haploid nuclei. Great, you're saying, what's the issue? The issue is that as the basidium matures, those 4 nuclei get packaged differently into the different spores. So the nuclei migrate into basidiospores. Only about 5% of the time, the basidia make homokaryon spores. These are inbred lines that we can use for breeding, and they contain 1 haploid nucleotype per spore. In this diagram, I'm coloring nucleotypes as either the blue color or the red color. And anytime you see like a little segment like this, means that there's been some kind of crossover event or a different independent assortment from the other parent. 95% of the time, 95% of the spores we get are heterokaryotic spores. And in the most common scenario, we call these N progeny. These are self-fertile spores, and they contain the parental type nuclei in the same spore. This is also where we get the name Agaricus bisporus, because instead of like other basidiomycetes producing 4 spores, our spores, we get 2 spores per basidia. Also, rarely, we get these S progeny, and this is where the sister nuclei from the meiosis get packaged in the same spore. And this has variable self-fertility. We don't know if there was a recombination event with a different mating type, which would allow it to be fertile or not, but this is a huge challenge because when you look at the spores that we can use for breeding that have some recombination, it's just these 2 scenarios out of these vastly more common scenarios here. A third challenge is a low recombination rate. So we have this meiosis occurring in the developing basidium, and there's not a lot of recombination. The recombination we do get is in subtelomeric regions. I pulled this figure from a 2016 paper, and it's showing the haplotype of a commercial white strain. So the blue represents the sequence that was derived from the U1 homokaryon H97, and the red is the sequence from the U1 H39 homokaryon. And you can see this is a commercial strain, and it's essentially the same as the U1 strain with a little bit of crossover events. We do have the advantage, I guess, that we appear to have normal independent assortment of homologous chromosomes. And this is what this is the end result here, where we get a little bit of recombination, if you will, or different parental material in these spores. But you can see that overall, these spores maintain heterozygosity. We have a lot of approaches to these life cycle challenges, and they involve a lot of manual labor. So we collect a lot of single spore isolates and then culture them up to mycelia. We're always looking for ways to automate this. And one potential way we can do this is by looking at differential or selective media to choose to select for or against haploids or dikaryones. We also screen mycelia visually. In general, the haploids they don't grow as fast as the heterokaryons. This is not always the case, however. So instead, we can use markers. We have molecular markers for mating type and cap color and some others around the genome. And this is very useful for screening. We also screen them to determine ploidy status. So from a bioinformatics perspective, where I'm coming from, this is pretty straightforward. We can look at the sequence metrics, alignments, and do some variant calling. But ultimately, our long-term goals are we want to increase those recombination rates and the frequency of homokaryon basidiospores. Some routes for this are using existing genetic variation for recombination rate. And again, if we don't have that, potentially using some molecular tricks or techniques that I mentioned earlier. Our fourth challenge is this challenge about mating types. So even if we do isolate our genetically unique spores and culture them up, you put them on a plate, they won't always grow together and create that hybrid. You'll know if you have a compatible reaction because you'll see the formation of this fluffy aerial mycelia that's dikaryotic. And this requires different alleles at the mating type locus. A 1994 paper by Kerrigan et al. Identified 8 mating types. And we have also identified a lot more mating types than just 1 or 2. However, you'll see the most popular mating types are mating types 1 and 2. And this is because these were the mating types that were found in that U1 strain that we made 50 years ago. We can use tester lines to determine mating types. However, we like to think we're, moving along and progressing. So these days we use PCR markers. The older markers were good at distinguishing mating types 1 and 2, but now we have better tools where we've sequenced different haplotypes, and we can use that sequence to make markers. This is work done by a research scientist, Daniel Steele. And we can see on this gel, it can easily distinguish a homokaryon containing just a single M1 band from, for example, a heterokaryon that has this M1 band, but also a mating type 3 band. Also want to quickly highlight some trait mapping work. So I know trait mapping isn't as sexy as it was like 20 years ago, but remember, we're still kind of figuring out this breeding thing. So the existing maps that were out there, they weren't really sufficient. And sometimes they're missing whole chromosomes. So we said, okay, roll up our sleeves, we can get this done ourselves. We did the regular way you would go about, doing a trait mapping study, assemble some diversity, sequence it, measurement. And here's just a highlight of some unfiltered data sets. Here, we found this QTL for cap color on chromosome 8. This has also been found independently by other research groups. In the future of mushroom research, I really think we need some more basic research to break this cycle. We need to look into meiosis and sporogenesis and figure out what is the genetic control of this packaging and what is wrong with us that we can't get the recombination that we see in other crops. Somoclonal variation is also, and epigenetics are also important because this is a clonally propagated crop. And so anytime you have this subculturing being done, this is immediately where my mind goes. The cellular composition and subgenome dominance are some aspects that we should also consider because we have these 2 different parents essentially existing in the same cell. So how does that affect things like expression? Is this different across tissue types or growth stages? Lastly, I'd like to test traditional methods. In the words of my former advisor, trust but verify. So the mushroom industry has been doing things for a certain way for a long period of years. And it would be great just to approach this with a new eyes. the future for our company, we're still all about variety release. We have a new white strain that's gaining popularity. We also have a very successful brown strain that's coming off patent currently. And we're always looking to make things more efficient through screening or trialing. For me, a genomicist, I think genomic selection would be a very interesting route to go down. Again, we also do basic research. We're scaling up our sequencing efforts. We're also looking at having some partnerships to help us tackle some of these challenges. And then having an open mind, thinking about alternative uses. What about metabolites, secondary metabolites? Can we market nutraceuticals more? Are there health benefits about that? And then using mushrooms for textiles or environmental remediation. And then not only considering the button mushroom, there's a lot of other mushrooms out there. Just being open to thinking about how can we breed those other systems. So thank you for having me. Here's some acknowledgements. I'd like to thank the graduate students and Corteva for this symposium. The engagement has been great. I also want to thank my coworkers. They work hard, they have good attitudes, and we all were working on this really frustrating but also fun crop. So thank you. Hey, Frank. The reason for calling it a hybrid is because essentially we have a haploid combined with another haploid, like our gametes, like our pollen, like in corn. But they live together in the same cell before, getting together. I do think that when I think of hybrids like maize, I think of heterosis, complementary gene action, things like that. And that is something that we have seen. So to have some different haploid, different nucleotypes coming together, that has been very productive for us. We have not done that to my knowledge. I think that would be very interesting to examine. Yeah, sorry, thank you. The question was, can we distinguish the homokaryon versus the heterokaryons by flow cell cytometry? Is that correct? Yeah, and that is something that's interesting. That's a good question. I don't know the answer to that. I think that would be something that we could definitely market. I think we would be very competitive in that compared to some of those other mentions. So he was asking about the efficiency of the mushrooms compared to chickens or other energy sources. But essentially I think we would come out pretty well in that regard, especially with, climate change friendly and like vegan alternatives being more and more common. Yes, so the question was, what kind of wild germplasm are we talking about? And what do they look like? There are some wild strains that are part of a collection. There was a big collection effort that happened in the 1990s. We do have access to a lot of those strains in our program. A lot of those strains are very small. So the size of the mushroom is very small, and they don't always grow in the even distribution around the surface. Furthermore, they open up very quickly. So they complete their life cycle from the mushroom development, and then they open up their caps and want to release their spores immediately, kind of like seed dispersal in other crops. And we really don't want to see that. We want to see a mushroom that is very slow and like holds on and doesn't mature that fast, because then we can, it makes it easier to ship it around, and the consumer gets a fresher mushroom. So yeah, the size of the mushroom and like the speed and their growth pattern. Yes, so the question was asking about the mycelial composition during the flushes of the harvest, successive harvests, and then also what do we do with the compost? So to answer the first question, we do see a little bit more colonization with each flush. So we do have multiple flushes, and with each flush, the mushrooms will come up. Again, a little bit in general, the pattern is like the first flush will be the most productive, second flush will be okay, and then the third flush. That's where like growers typically make their money, even though because these are a little bit tight harvesting, but the third flush will have fewer mushrooms, but they'll still be around. And after that third flush, we can use that compost and recycle that back into our production, because that's becoming a bigger thing with the scarcity of peat, and climate change, and trying to consider alternative composts. Since we are developing and like creating our own composts, it quickly adds up, and we try to have a very consistent product. But when we're making these huge batches, we have to be sure that we're testing them to make sure the percent nitrogen is within certain guidelines, and the percent, whatever is as consistent as we can get it. Yeah, so the question was, how do we prevent fungal pathogens of our fungus during the growing? And we have an IPM system. So we have, during the harvest, our harvesters will use gloves, and like we have a decontamination like protocol. So that's just maintaining like clean, practices helps. We also have a pest that is somewhat difficult because it can fly, it can move around. So there'd be like these small flies. If you have like houseplants, they'd be like fungus gnats. They kind of look like fungus gnats. And those can be vectors for some diseases, like on the caps or whatever, they can spread that. And so to control for those, we have like fly paper, like at the entrances and exits of our growing rooms. And that really helps cut down on some of that disease. Yeah, that's a good question. So the question was asking about how easy is it to transform mushrooms or use some of these molecular tools. It seems from reading literature, this has been done in other crops and other similar mushroomy type crops. For example, yeast is a fungus, and that's pretty amenable to transformation. And that paper, that 2016 gene editing study that came out, it seems like there was some potential there. But I think that from looking at the landscape, before we can really start, diving into that, we need to make sure that it's going to be consumer accepted. Mushrooms are already kind of, people feel a certain way about them compared to like plants. So, we don't want to shake the boat too much about that. So yeah, I agree. I think it would be really exciting if we could be able to use some of those tools. Sure. So the question was, how can we improve shelf life of mushrooms? So in terms of thinking of post-harvest factors, having a strong supply chain is one way you can do it. You can control the environment, making sure that you slow down the mushroom biology with cool temperatures. You can manage the humidity. And that has stuff to do with like the shipping and the packaging aspect. On the genetic side of things, we can also kind of modify that, where we want to breed mushrooms that keep their gills closed. They have their caps and they don't open up and mature, so they get all nasty and spores and stuff like that. There is genetic variation for that, and so that's like an important breeding target that we're looking at.
Next, I would like to introduce Dr. Sara Gonzalez. She is a marine ecologist and evolutionary biologist specialized in seaweed research, and particularly focusing on seaweed ecology, physiology, and chemistry for sustainable aquaculture development and climate mitigation. An alumni of Cornell and PhD from UC Santa Cruz. She is a postdoctoral scholar at Hole Oceanographic Institution. Her work centers on identifying heat-tolerant strains of sugar kelp and using satellite remote sensing to assess kelp forest health. Much like the wonderful person she is, and she'll be presenting her wonderful talk on not your typical crop, growing resilient seaweed for a warming ocean.
Thank you. Oh, can you hear me okay? Thank you, Vashti, for that wonderful introduction. And thank you all for being here. And a big thank you to the organizing committee for all your hard work putting on this awesome symposium. I'm excited to continue the theme of not plants, talking about some of the work I've been leading as a postdoc at Hole Oceanographic Institution over the last year and a half or so, where we are growing resilient seaweed for a warming ocean. So I did do my undergraduate degree here at Cornell, but at the time I was really mostly interested in animals and behavior and physiology. So probably the most plant-related thing I did was in my art of horticulture class, where I helped to build this sod sofa, which still exists next to Trillium. And I was really excited to get started in research as an undergraduate. I worked with shrimp parasites in the Chesapeake Bay, and spent a summer at Shoals Marine Lab, which is co-run by Cornell and University of New Hampshire, studying herring gull chick development and growth while avoiding being dive bombed in the head by their parents. But my world really opened up when I got the chance to experience seaweed for the first time when I did a Fulbright fellowship in Chile. And my research there was centered around kelp forest ecology and also kelp harvesting strategies and impacts of different harvesting strategies on the recruitment of invertebrates in the kelp forest. So I got the chance to travel around the country and speak to fishermen and kelp harvesters who were harvesting this resource. And I was just really impressed by not only the ecological importance of kelp for providing this foundational habitat for lots of fish and invertebrates, but also the importance that kelp has in human societies. Anywhere on the central coast of Chile, you could find kelp being sold in dried bundles at fish markets. So I was really excited to continue studying kelp for my dissertation work at UC Santa Cruz. And in particular, I was fascinated by the morphology of giant kelp, which was in that last image as well, and how it exists in these two different forms that if you dive down below to the holdfast, this structure that holds the kelp to the rocks at the bottom, sometimes the giant kelp would have this kind of conical, mounding holdfast, and other times it would have this elongated, flattened, stretched out looking holdfast. And I was just so curious as to how this could be. Is this an environmentally plastic trait, or is this something genetically fixed? And so I gathered spores from both morphs and brought them into the lab and grew them on these tiny pieces of string. And so they started out just a couple centimeters large, planted them into the bottom of the ocean, and 6 months later they had grown to be over 9 meters tall. And I was able to compare their morphologies and see whether this trait was actually genetically fixed or not. It's genetic, by the way. And it turned out that a lot of these methods I was using happened to be borrowed from kelp aquaculture, this seeding the kelp onto strings. So that transitioned nicely to my current work as a postdoc, and also this same motivation for wanting to understand the relationships between genotypes and phenotypes in kelp. But I want to back up and just talk about what is seaweed. So kelp is a type of seaweed, but here we can see that the seaweeds are a really diverse group of species. They come in all sorts of colors and textures and sizes, and they are categorized as large multicellular marine algae. And they are photosynthetic, but unlike plants, they don't have any roots or vascular system. And we group them as reds, greens, and browns based on the predominant color pigments that they have. And some examples would be nori, you might be familiar with, is often found in sushi rolls. Green seaweed example would be sea lettuce. And then the brown seaweeds are wakame, kombu, and other kelp, which you might have had in seaweed salad or in miso soup. So this is seaweed, but this is also seaweed. We experience seaweed in a lot of daily products that maybe we're not always aware of, but there are a lot of sugar extracts that we can take from seaweeds that have a gelling or emulsifying or thickening agent to them. We find them in heartburn relief medication, toothpaste, salad dressings, mac and cheese. And then in recent years, there's also been a lot of development in food products that kind of don't hide the seaweed as much. So in many countries, especially in Asia, seaweed has been a part of the diet for thousands of years. But in countries like the US, it's not as prominent in our diets. So there's been efforts to sort of mainstream kelp into things we're more familiar with, like burgers. And then also using seaweed extracts to make novel materials like bioplastics or fibers that can replace synthetic fibers and maybe start to replace some more carbon-heavy industries. And seaweed's also really sustainable in terms of its relative resource use per ton of edible protein. So in this figure here, we're probably all aware that beef is pretty resource intensive in terms of land, water, feed, and greenhouse gas emissions, followed by pork and then poultry. When you get into seafood, it's a little more sustainable, fish and shellfish. But seaweed is really the only crop that has the potential to be carbon neutral or even carbon negative, since it is taking up CO2 as it's growing. And it doesn't require any fresh water or fertilizer or arable land in order to grow. But okay, we're not going to all just eat seaweed. We still have other forms of agriculture, and that's okay. Seaweed can also help reduce greenhouse gas emissions in other forms of agriculture. Cows, we all know, emit methane, but there's actually a type of red seaweed that if you include in the cow's diet, due to the high bromoform content of the seaweed, actually can reduce the methane output of cows by 60 to 80%. In addition, we can just use seaweed as animal feed to have a lower carbon footprint than some of these other more common crops used for feed like corn and soybean. And if we can figure out how to grow seaweed at really large scales, we can start to tackle some larger issues like carbon dioxide sequestration. So as the kelp is growing, it's taking up CO2 like any other photosynthesizing organism. And then sometimes those kelp fronds may get detached, say during a storm, or bits of detritus are just sloughing off all the time. And that material eventually makes its way down into the deep ocean, where there's really not a lot of metabolic activity going on. So that carbon just sits there and degrades very slowly and is essentially sequestered for potentially hundreds of years. So there's some thoughts out there that maybe we could farm a whole bunch of seaweed, suck out a bunch of CO2 from the atmosphere, and then dump it to the bottom of the ocean. Just to give you a sense of the kind of scale that we would need to have a really sizable impact on CO2 sequestration, to sequester 1 gigaton of carbon per year, we would need about 730,000 square kilometers of seaweed. Right now, the total area of all the world's seaweed farms amounts to just 2,000 square kilometers, so we'd really need quite a lot of seaweed. And then there's also the question of what to do with this seaweed to sequester the carbon. There's some potential concerns about sinking this huge amount of biomass to the bottom of the ocean. What are the ecological effects of doing that where there's not normally this huge influx of carbon all at once? And it's also kind of a waste because we farm all this seaweed. We could turn it into something like construction bricks for buildings that could maybe hold that carbon in buildings that are going to last for a long time. So where does seaweed farming occur around the world? Over 95% of seaweed farming is happening in Asia, primarily China and Indonesia. But there's also a lot of other countries with high exclusive economic zones, which are the bright blue color there, including the US. So there's plenty of more ocean space where we could potentially be expanding seaweed. And seaweed farming is actually the fastest growing sector of aquaculture. And it can look different in different places around the world, depending on the species, resources. So sometimes you might have red seaweed growing kind of in shallow nearshore waters here. You might have really large scale red seaweed farms, some smaller scale ones in Faroe Islands and in Maine. And in the US, the major states involved in kelp farming are Maine and Alaska. And this species here, the sugar kelp, is the most commonly farmed species and is going to be the star of the rest of the talk today. So a lot of great products can be derived from sugar kelp, again, with those sugar extracts, for example, alginate. Depending on the particular species and actually where the kelp was grown, you get different compositions of these sugars, and that can lead to either more rigid or more elastic gels that can be used in different products like pharmaceuticals. The kelp can just be eaten straight as food. Might want to boil it down a bit so it's not so chewy. And then supplements like iodine are really popular coming from kelp. Biomedical materials like wound dressings and dental molds. And then again, as a gel or thickener for cosmetics and food. And because of its high nitrogen content, can also be a really great fertilizer. And even potentially extracting biofuel. Sugar kelp gets its name actually from the high content of the sugar alcohol called mannitol, which can be converted into biofuels. And we really do need a lot of innovative products to help mitigate effects of climate change. If you were on Earth last year, you experienced the warmest year in recorded history, and it's probably going to change at the end of this year. This is a map of the global temperature anomalies from last summer. And overlaying here, the sugar kelp wild distribution happens to be in a lot of these hotspots that are warming up pretty rapidly, especially in the Northeast US, where we have a lot of our wild sugar kelp populations. So sugar kelp is a cold-loving temperate species. They really start to degrade above temperatures of 20, 21 degrees Celsius. So warming waters are threatening sugar kelp aquaculture. It's increasing the vulnerability of those wild populations, which are the seed source essentially for our farmed kelp. It also shortens the growing season for growing kelp. It limits the time that you can plant the kelp because you may have to start waiting later into the fall where it's cold enough to plant, and you have to start harvesting earlier in the spring before it gets too warm again. So this is going to result in lower yields. And then warming also is restricting the latitudes where we can actually farm kelp. Luckily, there is substantial genetic diversity among our sugar kelp populations. And so our lab at Woods Hole works primarily with these northeastern populations from Maine down to New York. And so since there's this genetic diversity, it's likely that there are already some individuals out there that have adaptations to cope with heat. And so in this figure, the different colors represent different ancestry groupings, and each horizontal bar represents a different individual in these different populations. So if we can identify the individuals that have these adaptations to cope with heat and look at the genes involved, then we can really start breeding for heat-tolerant kelp. So before I get into how we do the breeding, we talk about the life cycle, and I'm glad we've already been talking about spores and such. That's going to be helpful. So what we do in our lab to breed the kelp is we'll collect the adult sporophyte, which is what we see growing out in the wild, and then usually running down the center of the sporophyte is kind of like a darkened band, and that's the sorus tissue. That's where all the spores are kept. So we can bring that back into the lab and pretty easily just release the spores, cut out a little piece of the sorus. That's enough stress to make it want to release all its spores. And then the gametophyte is the next life stage that grows out of the spores. So we have male and female gametophytes, which can come from a single sporophyte. And then under the right conditions, those will release their gametes and fertilize egg, which then develops into the juvenile sporophyte and ultimately the adult sporophyte. So in order to do breeding in the lab, we start by isolating a single gametophyte, put it in a flask of seawater. Over time, it grows into this kind of pom-pom, and then we're able to fractionate that up into little fragments. And eventually those grow, we fractionate more, and we end up with all of this biomass of fragments, all of the same genotype. So in our lab at Woods Hole, we have a collection of hundreds of these strains, all originated from single gametophytes. So when we want to do a cross, we can just take a scoop of this male gametophyte, a scoop of that female gametophyte, throw them together, and they produce sporophytes. So that process in a bit more detail, once we have thrown the gametophytes together and they have little microscopic beginnings of sporophytes, we actually filter that material down and then take a paintbrush, calligraphy brush, and paint that onto these spools of string. Then once those have a little more time to grow in the lab, we bring them out into the ocean where we wrap them around a thicker grow line. And so the kelp lines are just sitting out about 6 feet below the surface of the water. And then we deploy them in autumn, and then about 6 months later in spring, we harvest the kelp up onto a boat, and it's quite heavy, so we need some extra equipment to haul that onto land. And then we come back to the lab and we take a bunch of measurements to see which genotypes led to which kind of phenotypes. And we do see a pretty wide variety in yield, in length of blades, in density of blades, all just from crossing different gametophytes. And these were all grown within the same farm, a pretty small area, about 400 square meters. So we do see quite a bit of diversity. So mostly the lab has been working on breeding for high yield kelp. But we're also interested in breeding for other traits like heat tolerance. So our goal is to really breed more heat tolerant kelp to increase resiliency in the industry, to expand farming to lower latitudes and extend the growing season for kelp where it's already being grown. So the way we've gone about doing this is to start with the gametophyte stage. So we have our big collection of gametophytes. We first want to assess heat stress tolerance in these gametophytes. Then the idea is to cross from that heat stress experiment, identify gametophytes that we think are heat tolerant and ones that we think are not heat tolerant, and make crosses within each of those groups, and then grow them up into sporophytes and see whether our predictions at the gametophyte stage hold true at the sporophyte stage. Because ultimately what we're interested in is whether the sporophytes are heat tolerant. But if we can predict that sporophyte heat tolerance from the gametophyte stage, that's going to really help advance us in our breeding process. So I'll start by talking about the gametophyte heat stress testing. So we conducted this experiment in little tabletop incubators, putting the gametophytes in these 96-well plates. And I had 3 temperature treatments in 3 different incubators. So the first is the 12 degrees Celsius, that's their control conditions, that's where they're happiest. And then 20 degrees and 24 degrees represent current anomaly temperatures and future expected temperatures for the Gulf of Maine region, where most of these gametophytes originated from. So in each temperature, we had 93 genetically distinct gametophytes that we plated out with 3 replicates into each of the wells in the 96-well plate. And this is an example of what it would look like inside one of those wells, a bunch of little fragments, all of the same genotype. So we ran this experiment over a few weeks, and what we were measuring was photosystem functioning via chlorophyll A fluorescence. So probably folks here are familiar with chlorophyll A that mostly absorbs around 684 nanometers wavelength. So we used a plate reading spectrofluorometer to put our 96-well plates into there and measured the change in fluorescence of each well over time. So if we have a really healthy set of gametophyte fragments that are all fluorescing and we see high fluorescence intensity, we know that there's a lot of live cells there. If we see very low fluorescence intensity, we know that there are very few live cells in that well. So this was how we kind of tracked survival and health of the gametophytes over time. So let's see what happened with their photosynthetic performance. We started all the incubators at 12 degrees Celsius, gradually ramped them up to their final temperatures, and then held them at those temperatures for 2 weeks. So the y-axis there is the mean fluorescence intensity, and these are averages for all 93 gametophytes in each treatment. And we see that in the control, there was maybe a little bit of decline in fluorescence intensity. They probably didn't love living in a 96-well plate. We saw a much greater decline in the 20C and especially the 24C treatment. So the heat was definitely having a negative effect on survival of the gametophytes. But there was actually quite a lot of individual variation. So if we look now at the change in fluorescence from the beginning to the end of the 2-week heat treatment, We're looking at the percent change in fluorescence over that time for each individual gametophyte. And again, they had 3 replicates. So we can see that we span from some gametophytes that declined almost 100%, so basically fully dead, and others that declined 0 or even had a positive increase in fluorescence. So we're really seeing a wide array of heat tolerances here. And we had thought that maybe, since we collected from these 2 different regions, Gulf of Maine and southern New England, there's previous research shows a genetic break here. So we thought that maybe the southern New England kelp would have already some more heat tolerance adaptations, since they maybe experience warmer temperatures than Gulf of Maine. But interestingly, we didn't see any difference in the percent change in fluorescence between these 2 groups. So it really came down to just a lot of individual variation. And we were able to do a preliminary genome-wide association study based on that variation. We had over 900,000 SNPs, which are here on the x-axis. We have scaffolds, not chromosomes, which is why this axis looks kind of crazy. And then the y-axis is the relationship, or the significance of the relationship between each of those loci and the heat stress response. So we have not a ton of super significant SNPs, but we do see a couple peaks here where there are a lot of SNPs all in the same general region. So maybe this would be an area that we want to investigate further to see if there are any genes maybe coding for proteins that could be related for heat tolerance. We're currently working on that right now. Okay, so I want to talk about the next stage in this process. So we assessed heat stress tolerance in the gametophytes. Now we're going to see, how did our predictions go, and are they related to the sporophyte stage? Well, it would be really nice to test this on adult sporophytes, since we didn't want to wait that long. We used juvenile sporophytes, but we actually did also plant these crosses out to the ocean, and we'll be harvesting them in a couple of months. So we'll be able to look at juvenile to adult sporophyte relationship as well. Okay, so taking what we learned from the gametophyte heat stress response, we wanted to grab males and females from the top tolerant and the lower less tolerant. And so we made 5 crosses that we predicted would be tolerant and 5 that we predicted would be intolerant. But interestingly, kind of similar to the mushrooms, sometimes certain gametophytes just aren't compatible. And so we ended up with 3 tolerant crosses that produce sporophytes and 4 intolerant crosses that produce sporophytes. So how we did this was, again, taking that material, painting it onto strings, which we've wrapped around microscope slides here, and then gave them about 4 to 5 weeks to grow. When the sporophytes were about 1 centimeter large, we plucked them off from each cross and put each one into a test tube. And then we had an experiment in these incubators here. So the juvenile sporophytes were all in test tubes, and we had 2 incubators at 2 different treatments. One, again, control at 12, and a heat treatment that was gradually warming up to 22 degrees Celsius. We wanted to be sure to do a gradual warming so as not to heat shock the sporophytes and have them just all die, because we do want to see which ones are tolerant and which are not. So in each treatment, we had our 7 crosses, 3 tolerant, 4 intolerant, and 6 replicate blades, so 6 test tubes for each cross. And then in order to analyze how these sporophytes were either tolerating or not tolerating the heat, we took photos at 3 different time points. So first at the beginning of the experiment, then about 2 weeks in when the heat treatment had reached 20 degrees, and then about 3 weeks in when the heat treatment had reached 22 degrees. And the main metrics we were looking at were the total area, and then also the healthy area, which was calculated from the percent healthy tissue. So we took these images and analyzed them in ImageJ to get the total area. And then we created an algorithm that would characterize each pixel in the blade as either healthy or degraded based on how much blue light was being absorbed or not. So these images were taken over a backlit surface. And we said that if more than 50% of the blue light was coming through, that means there wasn't a lot of chlorophyll there. So that's a degraded tissue area. If less than 50% of the blue light was coming through and was being absorbed, then we know that was healthy tissue. So for example, the algorithm told us this blade was 97% healthy and this blade was 36% healthy. And from there, we just multiply by total area to get the healthy area. So let's take a look first at individual cross variation for total area. So we can see that in the heat treatment, most of the growth, most of the growth was happening, over this first time period. And then they were sort of just maintaining the area that they had amassed. Whereas in the control treatment, we saw more of a steady increase throughout the duration of the experiment. And a kind of similar trend with the healthy tissue, although some individuals in control were also losing a little bit of healthy tissue in that time. But so really most of the growth of healthy tissue happened in that first up to 20 degrees and then more just maintenance up to 22 degrees. So now let's see what happens if we group them by their prediction categories. So we can clearly see that the left graph here is the mean % change in total area from the beginning to the 20C mark. We see that the predicted tolerant groups, or the darker bars, had a much greater % change in area compared to the predicted intolerant group, and a similar story for % change of healthy area. We see that the predicted tolerant group had amassed more healthy area than the intolerant group. And importantly, we see that there's not a difference between the two groups in the control treatment. So this means that our predicted tolerant group was able to grow more and more healthy tissue in heat without compromising their growth in cold conditions. And this is important because on the farm, it starts out cold and gets warmer, so you want the kelp to be able to grow well in all of these conditions. Now, when we get up to 22C, we see that the tolerant group is still maintaining more area than the intolerant group, but it's not necessarily healthier. There's no longer a significant difference there. Although we did have kind of a small sample size, so possibly with more samples, that would be significant, but it's possible also that this tolerance just didn't hold up to 22, which is really quite stressful for the kelp. So what we've learned so far is that our gametophytes display a wide range in heat tolerances, even from just this relatively small area from Maine to New York where we've collected from, and just looking at 93 gametophytes. So this is really promising that we have a lot of variation to be able to select on. And we know that the sporophyte progeny of our predicted heat-tolerant gametophytes did grow bigger and healthier than the predicted intolerant sporophytes, at least up to 20 degrees Celsius. So this is really key, and this is one of the first studies that's really relating gametophyte phenotypes in kelp to sporophyte phenotypes. So making that connection is going to be really important moving forward for heat tolerance or other traits too, looking at alginate sugar content, mannitol, other desirable traits for aquaculture. And we're even able to see that there may be some specific regions of the genome that could be related to heat tolerance. So we're currently conducting another study, another round of 90 gametophytes to add data to this set so we can have a more robust genome-wide association study and maybe see what those gene regions may be doing. So I think we've made a few steps closer to our goal of producing heat-tolerant kelp to increase crop reliability, extend the growing season, and expand kelp farming to warmer latitudes. But of course, always more work to be done. And I want to really thank everyone who contributed to this study, folks in Scott Lindell's lab at Hui, as well as our collaborators, including Jean-Luc, who's here in the audience, and all of our funding sources. And thank you all again for being here. I'm happy to take any questions. Yeah, that's a really great question. The regulations around that vary a lot by state in the US. So for example, in Alaska, they have a rule that all the kelp that you're growing has to be sourced from kelp within 50 kilometers of your farm and represent at least 50 genetically unique individuals to try to preserve that diversity. And Maine also has strict rules about if you've taken kelp out of Maine into the lab, it can't be planted back in Maine. Then some other states that haven't had kelp naturally growing in a while have a bit more laxed policies because I guess they figure, well, if kelp grows there, it's not going to interfere with the existing population because there really isn't an existing population. So it does depend a lot regulation-wise by state. But I think biologically, we still need to do more research, particularly each individual species, to understand what kind of is the probability of interfering with the local population. How much diversity do you need to ensure that you won't have single genotypes kind of taking over? I think a lot of that research still needs to be done, but definitely a concern on regulators' minds. Yeah. Oh, great question. So we keep the gametophytes under red light to keep them in a vegetative state. So that prevents them from becoming fertile until we're ready to cross them and induce fertility. Yeah, yeah, certainly. So the first question was, sorry, can you say the first question again? How do you go ocean space? Right. So in our lab, we mostly work on just pilot scale farms, so really small, and they're mostly in coordination with either existing kelp companies that have leased the space and they want to test out certain farm designs or see how different strains work. And so we've made partnerships with companies to use their space or universities that have that we can work with, and we get permits for just these small scale farms. But yeah, there's a lot of other ocean users, particularly in nearshore areas, fishermen, shellfishers, although sometimes we also do work with shellfish farms, and there's some work also trying to integrate growing seaweed next to shellfish for kind of mutual benefits there. So at larger scales, there certainly could be impacts of shading. So I think it would be important to choose areas that maybe like have sandy bottom where you don't have a lot of things growing there anyway, or maybe do kind of smaller patches of farms, leaving spaces for light in between. There's lots of study on engineering and design of these farms to be able to accommodate existing sea life. Thank you. Oh, interesting. Yeah, so we did look at some in a separate study I didn't mention here with adult sporophytes, looked at interaction of heat and nitrogen content, because there's been some studies that show that with increased nitrogen, maybe the kelp is able to sort of overcome more heat conditions. We didn't actually find that was the case, and so there's been other work that shows for other species that that isn't always the case. So I think it is really region and species dependent whether the extra nitrogen is helpful. As far as acidification, generally more CO2 the kelp kind of like, but there could be indirect impacts by impacting the other life around them that may have some other factors. Yeah. Okay.
Have you looked at the practice?
No, we haven't. We definitely see that on the farms, as it gets closer to summer, there's a lot more fouling on the kelp. So there probably is a relationship there, which as things are warming up, those organisms become more abundant in the ocean, perhaps. So there probably is also a relationship with other disease. Yeah, we've not looked into that. Okay. I don't know if maybe Jean-Luc can offer. No, yeah, I don't think we have really any ideas what's going on. It seems really, really random at this point. Have you found any particular? Yeah. Yeah, I mean, I guess because normally in nature, an individual sporophyte has like millions of spores. So if one doesn't work with one other one, it doesn't matter for the reproductive success of that sporophyte. Yeah, interestingly, we didn't see any correlation between heat tolerance and the populations that these were sourced from. So we did look by region and then also by population, and there wasn't a correlation there. So it's really amazing how much diversity we saw within populations and then sometimes within a single sporophyte. So sometimes we had essentially sibling gametophytes that were sourced from the same sporophyte that would sometimes also have different heat tolerances. But also important to note, these gametophytes that I was testing, they were originally sourced from sporophytes collected back in 2018, and they've been in a cold chamber storage basically for 5-6 years. So it's possible that through cell division in the cold chamber, they've reacclimated to all kind of be at the same baseline, maybe. Okay.
So it's not quite like chromosomes and female that dominance is similar in health.
Oh, dominance of certain, yeah. Sorry, I don't think I totally understood the question. Mhm.
Do they just coexist in the cell?
So the gametophytes are actually a free-living life stage. So once the spores get released, they develop into the gametophytes, and the males and females are separate organisms essentially at that point. And then the female will grow an egg, which the male will fertilize. And at that point, then the sporophyte grows from the egg. Was that your question?
Yes. Diploid?
It's diploid at that point, yeah.
Doesn't make sense to think of it as chromosomes.
We had representation from about 11 of those 15 populations I showed. So pretty good representation along all of the groups. Yeah, didn't look specifically. That was just, yeah, didn't look specifically at ancestry groups. From the 2 regions, that was from the gametophytes. No, those were from our gametophyte collections. So they were collected from wild sporophytes. But when I showed the 2 regions that were not different, that was based on the heat tolerance assessment of the 93 gametophytes from our germplasm that have been harvested and cultivated essentially in vegetative form. Mm-hmm.
In wild, would they also last that
long? Probably not. The kelp adults can last more than a year, though not typically. But there is some research showing that the gametophytes can actually kind of stay dormant in the sediment, I guess, in case conditions are too warm or otherwise not favorable, and then become fertile when conditions are better again. But I don't think it's years. I think that's more like on a month's time scale. Yeah, so it is kind of unnatural the way we're keeping them for sure. Yes, question back there? Or no? Okay. Yes. Kelps without sugar? I don't think so. No, I think they've all got either some form of alginate or agar, and the red seaweeds have carrageenan. Yeah, but the sugar kelp just has more, I guess, than other kelp species. Yeah, I don't know. I don't know if we have that. But I mean, it's not like glucose sugar, you know, it's a different form of polysaccharide, so it doesn't really taste sweet. Yeah, that's an interesting point. So at least in the US right now, the structure of farming, particularly on the East Coast, is that there are a few kelp hatcheries that have the kelp seed, the gametophytes basically, and then farmers can buy seed from there. So I guess the heat tolerance breeding or any sort of breeding would really happen in those hatcheries. There could potentially be farmers who want to breed their own, but typically they're buying from larger hatcheries. And so the idea is that maybe if we can identify genes related to heat tolerance, it'll be easier for hatcheries to sort of screen their collections to see if they can find individuals with those traits.
We have our final speaker for the symposium. Kevin Folta is a professor of horticulture sciences at the University of Florida, where he researches photomorphogenesis, controlled environment agriculture, the genetic basis of flavor in fruits, and the control of flowering. Dr. Folta is also an experienced science communicator and has hosted the Talking Biotech podcast since 2014. He's been recognized with the NSF Career Award and the Cass Borlaug Award in Agricultural Communication. Today, he will highlight work on breeding crops for controlled environments in his talk titled Controlling E, Controlling G, New Crops for Controlled Environments. Thank you, Kevin.
Well, I guess I have to start out by admitting I've got organism envy. I mean, you got like Jaffa mallow and hairy vetch and sugar kelp and mushrooms and I got lettuce. But I do weird stuff to it, and that's kind of the take-home message. Is in thinking about the presentation today, which is most appropriate for perhaps a younger audience for a student organized consortium, is when I was in your shoes, I was thinking, you know, everything's pretty much figured out already. What am I going to do? And I think what happens is you start to find funny avenues of research to open up, the more you think about ways that we can think of useful application and also new ways to solve old problems. And I'll be the first to admit, I'm not smart as most of the people I work with, but I'm clever and I work hard. And I think you'll see that's kind of the theme here. There's different ways you can solve a problem and come up with new technology. And I think that's what we're going to cover. So today we'll talk about genetics and environment. We'll talk about the way these things work together. So my laboratory, it does a lot of stuff. We historically have worked with strawberry genomics. We were the laboratory that led the sequencing effort of the diploid strawberry way back in 2010. And it was the 12th plant genome sequenced with no physical map and all short read assemblies. Pretty cool stuff back then. And then we do some stuff with light. And then the best project is how we wreck things, plants and microbes with random peptides. So we find lethal peptides that kill things. It's really kind of neat. And then New Crops for Florida was kind of born through COVID. We're experimenting with ways to grow, find new crops that can work really well on small farms, and really exploring the universe of novelty to see what we can do for small farmers to introduce local consumers to things they've never tried before and things that do very well. And it turns out that's actually been really exciting work, mostly because in my when I'm not at work, I'm at home at work. I got married to a farmer, and she was a subsistence farmer in Ukraine who came to the U.S. to my town, and now we do a lot of stuff. We grow seasonal fruits and vegetables. We also do turkeys and pigs and chickens and eggs and all. We sell at the farmers market every Saturday, except I won't be there tomorrow. And then last year at 56 years old, I started a new kind of breeding project. I got an F1. That's kind of cool. She'll be at the market tomorrow. She's the main attraction. So I get to not only research in fruits and vegetables and how to grow them better, I also get to do it and expose consumers to things they've never tried before. And it's really exciting. It's a lot of fun to be able to do both things. So what we'll talk about today is how we can think about G and E in creative ways. So one of the big problems with controlled environments, and we get excited about controlled environment companies that think of all the good things they can do. We can repurpose the infrastructure in urban areas. We can train an urban workforce for high-paying, okay paying, highly skilled jobs that you can move up inside a company. I've been up for 36 hours, so I'm blanking on words. Short supply chain, it goes right to the consumer down the street rather than across the country. Low carbon footprint. We get water savings. All these things are really, really strong suits of controlled environment agriculture commercially. The problem is the amount of energy that's needed. So can we think of clever ways to invent varieties that will do better inside that controlled environment? And can we come up maybe with new practices, new ways to deliver that energy that can make these things more viable? And I've worked with a variety of companies over the years and in different capacities. All of them have gone out of business. So I didn't do much for them, I guess. But this just shows how they're how there is a need for thinking about this differently. And that's what I've always kind of brought to the table. That kind of is why they invited me in, I guess. The other part of this is that I've gone to a lot of meetings over the years in controlled environment agriculture, and it really stirred the pot because I've come up with seemed like radical ideas, like just don't grow them under a pink light, change the lighting as plants change through development. Don't just grow them under light, control how they grow. We invented the concept of plant whispering. Can you give plants a vocabulary of light signals to control growth and development? And just it seemed like such a better idea. And that's kind of where we are now. And also the idea that, which seemed kind of silly, but was really important. And what we'll talk about today is don't just stick any plant from the field into the box and expect it to perform. Just like any environment, you have to breed for it. And I'll show you how we are starting to think about that today. And we know that plant growth and development plant productivity is really a mixture of genetics, environment, and management. And that how those three things align provide us with some sort of useful commercial plant output. And the important message in this is that you have some certain set of genetics that you can only stretch so far by changing the environment. The genetics are constrained by the environment that they're in. And in the field, only thing we can control are the genetics and the management. We're at the mercy of the environment, which is really one of the main problems. We're at the mercy of the environment. Okay, and then in a controlled, closed environment, such as these operations with artificial lighting, we can control management in the environment. And now we need genetics that fit. And that's the argument that I've always made, because I think the problem here, oops, where are we here? The problem is this, same as this. It's like trying to win a dog sled race with dachshunds. You've got a breed that was not made for that environment, not performing well in that environment, and you're the guy on the back of the sled saying, what's wrong, right? You need to have a breed that's made for that environment, that likes the cold, likes the snow, likes strong, is excited by exertion, that kind of thing. So you have to have the right variety for the environment you're using. And to a lot of folks in the controlled environment space, this was like an insane idea, because a lot of this is all dictated by folks who are in the technology and lights and things like that. They forgot that the most complicated machine in that room is the plant, and radically underthought that part of the equation. And so I think these ideas we're starting to introduce now. So here's the crisis and opportunity. So what are the things that we breed for in the field? What are some important field traits? And yield. Okay, what else? What else? What else to consumer? What else? Yield, all right, yield. Yeah, we love that yield, love lots of it, yeah, yes. The timing of flowering, right? Is it an appropriate plant for your long days or short days? What else we got? What was it? My ears are ringing from lack of sleep. What was it? Pathogens and disease, pest and pathogen resistance, right? And maybe some other stuff like, water resources, there's tons of things that you need. Shipping. Can I send this from California to Ithaca, New York, and have it still show up in a pretty decent state, right? So the priorities for the field are things that benefit the producer, not necessarily the things the consumer wants. So if we can control that environment and have a more locally set, what are some of the things we could breed for? Flavor and aroma, right? What else? Appearance, make them look better, make it so the kids want to eat their veggies, right? What else? What was it? Lower inputs, right? Things that survive on maybe a sprinkle of nutrients that we can control precisely through hydroponic or other types of means. And then precise amounts of water, which is becoming an increasingly scarce resource. Maybe things that don't require pollination, maybe things that are seedless. maybe things that, still have good yield. You can think of all of these different qualities that would lend themselves very much to growth in a controlled environment. And I give some ideas here, because here, if we can control the E, it means that we can play with G. And that means we can have a whole set of new priorities. And as we are going through the idea of field priorities, it was yield, 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 right? Disease resistance, these kinds of things. Can you ship it? Post-harvest quality. But if we have the opportunity to breed for the specific environment because we can constrain E, now we can look for things that maybe grow rapidly, get out of the box fast, get something that has that lends to fewer inputs, reduce plant stature, ease of harvest, high crop value, use of less energy. We'll get to that. But then novel flavors and colors when you induce the flowers. Can we flip a switch and make the plant flower? Think of how much money that would save for the cut flower industry. Being able to stop flowering. Things like basil, lettuce, shut it down. Manipulate nutraceutical value, not need pollinators. We don't have the same disease pressure. It may be different or maybe none at all. Some of the new places are growing plants for medicinal production. Do this in clean rooms with absolutely no pest pressure. Do this in spacesuits and all that good stuff. And one of my favorites is plasticity. Multiple phenotypes from one genotype. Can we flip the switch and produce a slightly different product? And I'll show you some evidence of that later. Pretty cool stuff. It all boils down to this idea is that when you're breeding for the field, plant breeders want an exceptional cultivar is one that is always good, no matter what happens in the environment. Because you're going to have cloudy days and rainy days, you're going to have sunny days and drought, yet your product that comes out is always going to be acceptable with maybe some minor times when it's not. When you can control the environment, you can control that trade presentation so that's always optimal. And that's what's exciting about this kind of approach. Can you breed something that fits this particular spot? And can we develop Energy Star plants? And that's the thing I've been thinking about as someone with a background in photomorphogenesis. Can we develop cultivars that wisely invest their photons, that turn signals from the ambient environment into more immediate developmental change, get them fast out of the gate? To the industry, a day is money. So if you can make it grow faster in the early development, that translates potentially to millions of dollars at the commercial level. So if we were to do that, are there some ways that we could just take a look at the existing germplasm in any given crop and start to make decisions about which variety we may choose because of its ability to integrate environmental signals more thoroughly? And that was the hypothesis we went to test. And it was based on the idea of the fact that plants have multiple photosensory systems, different systems that sense the red, blue, ultraviolet, near UV, far red, and even the green portion of the spectrum, and tailor physiological responses. And these are all mediated through different photosensory pathways. And could we identify varieties that have been selected because they had a better response because of throughput through these individual light sensing pathways? So in other words, is something more sensitive to red more likely to give us a better yield? And looking for those associations between early and later growth. And that's what we're working on now. But the big deal is that you have all of these different input channels that we can monitor very easily in very rapid experiments at the seedling level to begin to ask questions about potential plant productivity and quality down the road. So we think about Arabidopsis. Talk about going back to organism envy, the most mundane critter on the earth. But Arabidopsis is great because we know a lot about it. So these two Arabidopsis seeds are, well, these two plants, two seedlings, are siblings from the same silique, one next to each other in that little seed pod. And the difference is that they have radically different morphologies. The difference happening because one was grown in darkness, one was grown under complete light spectrum. And you can see some significant changes. An inhibition of hypocotyl elongation, expansion of cotyledons, development of chloroplasts and chlorophyll, and a little root elongation thing going on there. So these differences were brought about because of that environmental signal. I always like to show this in schools. I always say it's like having twins, and one of you goes in the closet for 10 years and comes out taller, you know. Kids get that idea. So if we were to look at some of these cues that light brings about, such as changes in hypocotyl elongation or cotyledon expansion, could that give us hints as to certain varieties that may be more suited for growth in a controlled environment because they integrate photons better? And that's the hypothesis. And the lettuce is kind of neat because there's a lot of different varieties of lettuce out there, and lots of variation in color and other qualities. It's highly homozygous, so it gives us a lot of opportunity to later on do very sophisticated experiments to identify causal genes. And it's easy to do these experiments on Petri dishes in 96 hours. So instead of growing fields of plants to identify, it's a lot like marker-assisted breeding. The only thing is we're looking at markers which are morphological. We're looking at how the seedling behaves rather than looking at a PCR product or a DNA sequence. We also are working on automating this to be able to do massive numbers of seedlings at one time. So that's something else that's coming. But what the main hypothesis is that if we see hypersensitivity in a seedling, it may translate to efficiency in mature plants, or at least be able to get to be a mature plant faster. The other part of this is that could we look to hypersensitivity to blue or red, far red, and be able to breed these things together to get all of the hypersensitivities in one place at one time? And the other part of this is that could we really make an ultimate plant for a controlled environment that may do very well in low light, significantly fewer photons, to get the same output, because all of the developmental channels were primed. I'm kind of a old school disciple of Fritz Wendt. I really believe that, yeah, photosynthesis is important, but setting the stage for photosynthesis is even more important. And getting the plant to have a morphology that's more conducive to handle the incoming light is even better. How do you make a bigger antenna? That kind of thing. And that's how we can maybe be able to grow plants with less light energy coming in. And so this is an example of how this works. And this panel over here is much better. These two are the same. I don't know how that happened. Over here, it gives you an idea of butter kings, both grown under dim blue light. This one very, very hypersensitive to light, and this one very hyposensitive to light. If I showed you dark grown, they'd look much more like this one here. So there is just inherent variation that you see across lettuce. And we're currently putting together a paper where we've looked at over 100 different lettuce varieties under different spectral conditions. And you see this massive variation that's there that tells us a little bit about how it will respond to the light environment. We also have some hints, and I'll show you one in a minute, about how what we see in a seedling does translate to more mature plants as they grow. So there's this huge variation that we can exploit when we look at this. And I'll show you one really nice example. Cade Cooper is an undergraduate in the lab. He does a really, really nice job. So down here on the bottom, we have just regular oak leaf lettuce grown under red light and blue light and white light. And I didn't put how much. It's 50 micromoles per meter squared per second for those of you who enjoy that stuff. So just kind of about 3 times room light. And if you look at this Thai oak leaf accession that is available, you see it doesn't quite expand the same way under white, looks about the same under blue, and under red has very long hypocotyls and very small cotyledons. Almost like it's not seeing light. It looks a lot like those dark-grown Arabidopsis seedlings. So what this is telling us, perhaps, is that when you're not seeing red, it's signaling a deficiency in the ability to transduce red light signals through phytochrome B, one of the red light sensors. And it's consistent with a lesion in phytochrome B signaling. What's really cool about that is that we also know phytochrome B has some other problems. It also shows a little hint or hypersensitivity to shade. So shade avoidance responses are when you move a plant from full sunlight into shade, the spectrum changes. You see an inversion of the red to far red ratio. You have much more far red than red, and also a significant amount of green to red, green to blue. And that shift causes plants to take on a shaded phenotype. So that rosette plant that typically is flat on the ground will begin to have exaggerations in leaf inclination and leaf expansion. So it tries to reach above its neighbors, sensing that it's close to, has close proximity to neighbors, or maybe being under a canopy. So those morphological changes occur to help it get up above. So if this is a phytochrome B mutant, we would expect it to have that same sort of exaggerated rosette phenotype. And we would expect it to be extremely exaggerated if we change the red to far red ratio. And that's exactly what we see. Well, those were the quantitative data for the previous thing. So here is regular oak leaf lettuce, just in red with a very strong, well, an even red to far red ratio. Here's another type of lettuce, red to far red. And here's that Thai oak leaf. And as predicted, it has that exaggerated shade stress phenotype, even though it has plenty of red light. It doesn't appear to be in the shade. It thinks it is in the shade because its photosensors are out of whack. If we elevate the far red and decrease, well, if we change the red to far red ratio to favor far red, we see that exaggeration in the other varieties, an extreme exaggeration over here. So this was kind of was consistent with the idea that we could look at seedlings and begin to make predictions about how mature plants would grow. The funny part about this, the interesting part about this is that we went back to say, well, what's going on with this Thai oak leaf lettuce? What do we know about it? Where does it come from? Why was it selected? And there's very little information known because really only a couple places sell it. But it turns out, oh, before I get to that, sorry. It turns out that phytochrome B, where we saw the lesion in red light sensitivities. This is Arabidopsis under red light, wild type, and 5B mutant. This looks a lot like that Thai oak leaf seedling under red light. And it turns out phytochrome B is a thermal sensor. It helps with constraining the response to heat on warm nights. So phytochrome B is working presumably to tailor plant responses to heat. And a number of papers came out in Science and other places that really show this very well. And so we may predict that Thai oak leaf, selected for a long hypocotyl, may be more amenable to growing in warm conditions, especially warm nights, like we have in Florida, like we might have in a controlled environment, right? So it turns out that if you go back and look at why was this thing selected, where did it come from, who did this? Thai oak leaf lettuce, loose leaf lettuce here, it says grows pretty fast, but excellent quality lettuce under high heat, a favorite. Resembles oak leaf, but with larger, more tender, and more upright leaves. So all of these things that we figured out backwards by looking at seedlings and analyzing behavior were things that they already knew. And it was the basis of selection for this variety. So it really validated our approach. And if we would have just paid attention to the website we ordered the seeds from, we could have saved ourselves a lot of time. But it validates the approach. It says that we can use phenotypes to potentially learn something about the long-term growth and developmental characteristics of a crop plant. And the reason this gets exciting is because in Florida, lettuce is a significant industry. We grow lettuce in the winter. The problem is when it gets warm, some things happen. We get bolting and poor growth. Here you have a lettuce that's heat tolerant and shows moderately slow to bolt. So what this suggests is that if we want to accelerate lettuce breeding for Florida, we could potentially be growing them under red light, looking at seedlings and automating that process, watching seedlings grow through time and finding the ones that grow fast through red light. And when we make crosses between heat tolerant varieties and maybe those that are not, using the phytochrome B deficiency as a way to signal which ones may give us better ability to grow in the field. And then we can test that. And that's where we are right now. We're currently validating a number of varieties and a number of crosses to see if that hypothesis is supported. Pretty cool stuff. And the big deal with that is that we aren't just constrained to lettuce. This is phytochrome B. This is a photosensor that's ubiquitous. And could we potentially be looking at any particular seedling to identify heat insensitive or maybe better heat coping varieties by looking at the seedlings which don't grow short under red light? And then, of course, what does that say in the long term about flowering and productivity? There's other caveats, of course. Some things we don't want to flower. Some things we don't want to bolt. Other things it's pretty important. So it's going to depend on the specific crop. But it goes to an interesting idea that we can use a seedling to tell us about energy consumption. And right now, especially as climates are changing and things are getting warmer, the vegetative crops like your basils and your lettuce and those things, if we can look at the seedling level to begin to make selections, maybe it can go a lot faster. And the other point on this is in a controlled environment. Some of the big expenses, of course, are energy. Something like at least a third, is some estimate as high as 60% of the budget of a closed controlled environment is in lighting it and then getting rid of the heat from the light. If you could keep things a couple degrees warmer by growing crops, which maybe grow a little bit faster, but also are insensitive to the heat or better able to handle the heat, perhaps this could be another solution for the controlled environment failure problem. The other thing that's really important is I think this idea of controlled environments, another opportunity in the crisis is, can we breed for plasticity? And this is another neat one that we worked on, was examining lettuce varieties for those that could give us the widest array of phenotypes from one common genotype. So we talked before about the idea of genetics and environment. Could we put the same set of genetics in two separate environments and get two different products? So in other words, a grower would only have to buy one set of seeds, understand the management around one set of seeds, and then test the market and say, what do you want, purple or green? And then push the switch that gives you purple or green. It's another aspect that hasn't been exploited yet by controlled environment agriculture, but an important part of plant whispering that I think that is something that should be adopted. I think the idea that can we grow a variety of plants from start one set of plants and then divide that up based upon the demand from the market. And don't pay attention to this on-off stuff yet. I'll get to that in a minute. But this is one variety of lettuce that already exists where we can make three very distinct products just by how much we turn the light off. So we're investing darkness to change the product and potentially put more kilograms per bag, or well, more biomass per bag by investing less in energy. So as a grower, this is a good thing. You're costing me less money, but I'm getting a more valuable product. And so maybe that's another way that we need to think about this. And we'll come back to this in a minute. This next picture I'll show you shows you how when we take one variety of lettuce that we've been working with, we can place it into 6 different light conditions and generate 6 different products. So that's 6 different products that are coming from one set of genetics. So 1 G and 6 E's. Well, taking that one set of G and pulling it in 6 different directions to make 6 different products. And it's another way to think about how we can better exploit controlled environments. And right now we're looking at herbs. We're doing a lot with this because we've done some interesting preliminary data. We have some interesting preliminary data on the flavors and aromas of basil, and shown that when we put it into a controlled environment of different spectral quality, we can radically shift the components that lead to liking and not liking. And this is old published work at this point. And the thing I want you to take home, so what we're looking at here are the major classes of flavor compounds, and then how they look in a greenhouse. So you're looking at less stuff and more stuff as you go from green to purple. Less of this, and and then shifting as we go up into more complicated narrow bandwidth environments. And it really shows that there's a tremendous opportunity to, just as you're pulling colors in that salad, be able to pull at the molecular circuitry that underlies the production of flavor and aroma compounds. And what was particularly interesting about this, that we learned something really cool about this. When I teach my plant physiology classes, I talk about how plants adapt to a new environment, how that's why they survive, that they have to be agile and adapt to the new ambient environment. That's why they do what they do. We have really nice data that show that once you're born in the greenhouse under a full solar spectrum, you remember that you were born under a full solar spectrum, even if we do funny things to you later. And if you were born under LEDs and an electronic canopy of narrow bandwidth light, and we move you to the greenhouse, you remember that you were born under funny lights. And this shows that maybe we need to think a little bit more about the way we treat plants early and how we can shift them to different environments later on. There's a lot more layers to controlled environments than simply putting them under a pink static illumination. And I think that's another trick that, as we move forward and become more sophisticated in our treatments, may make the idea of controlled environment agriculture increasingly more viable. So how do we, and this is always the part that I like the best, how do we do something weird? How do we break biology or enhance biology by doing something that nature never intended, by taking something that evolved under on an evolving planet of dark and light, of a solar spectrum that's complete, and now we throw it into an environment that could never happen on Earth? Could we get something beneficial? Or better yet, could we learn something that could inform the development of new practices that allow us to save energy? And this is kind of neat. I think this is kind of fun, and it's working well. So what if we were to take plants and instead of breaking up the day into 12 hours of dark and 12 hours of light, we said, let's give them 8 hours of dark, 8 hours of light, 3 times, and 6 hours of dark, 6 hours of light, 10 minutes of dark, 10 minutes of light. What if we broke up the day like that? And we have some specialized equipment that's homemade that does this. And what's really neat about this, and what you might be thinking is, well, Folta, you're an idiot because it's the same number of photons in both cases, the same amount of energy, still 12 hours on, 12 hours off. And you're right, but I'll show you how this gets really cool. So if we look at, let's just look at hypocotyl elongation. This is, these are seedlings of different crop plants, or seedlings of kale, turnip, and beet, all of which are used as commercial sprouts. And let's stay with hypocotyl elongation. What you see here is that if we go 12 hours on, 12 hours off, you see this nice suppression of hypocotyl elongation that says you're getting a lot of integration of light signals. And if you start to mess with this, we'll stay up here in kale. As you start making that interval shorter and shorter, the plant appears to be more dark-like. So it's getting the same number of photons, but as you break it up, means you make the photomorphogenic integration of those signals less correct. Until you get down into the 5 seconds on, 5 seconds off range. So you start getting into the range down here, you're actually doing a really nice job when you start getting in the 5 seconds on, 5 seconds off. And that's kind of interesting. So all of a sudden, the plant is thinking that it's under constant illumination again. All right, so let's just keep that in mind. The 12 hours on and off and the 5 seconds on and off appear to be the same, where the rest aren't. And we wrote this paper. Well, there's more to it coming up. But we wrote this paper. It's all done. And the student who was in my lab, he was an undergraduate. He was awesome. And he comes to me and says, oh, yeah, hey, by the way, this is all good, but somebody already did this. And he's actually a professor at Wageningen now. He's a good guy. He says, somebody already did this. And it turns out that he went back in the literature and found a paper from Garner and Allard in 1931, or Garner and Allard did the same damn thing, only with Cosmos and some other critter, probably, I don't know, something else, larkspur, whatever that is. But the idea was, as you grew them under different intervals of dark and light, and I love these old things. Fritz Wendt did a lot of this too. When you just put plants in funny environments and saw what would happen. But here they were doing those treatments, 12 hours on and off, 1 hour, not so good. And down to 5 seconds on and off, it comes back. So it's like almost the same experiments that they did 80 years previous and looked very similar to the data that we got on something like kale. And that's anthocyanin accumulation, which could also be used as a marker of photomorphogenic development. So the coloration that we see in the stem started high, and it was really, really high after 5 seconds on off. Okay, so now we go back to, so why is this happening? Why is short not bothering the plant? And what this shows is the complicated mechanism that's entraining its circadian oscillator. The internal oscillator of the plant is what's measuring that day length and reporting to the nucleus to coordinate gene expression so that gene expression is appropriate and anticipating the next light cycle after a certain period of darkness. And this has been very well dissected. I think what's happening, and we're testing this now, is that when we give these short little pulses in the beginning of that seed to germinate, it never really conditions a clock. The oscillator never is moving in any particular direction. So we kind of are bypassing the oscillator and just running the plant strictly upon input signals from the small pulses. And that when you're at 12 hours on, 12 hours off, you're running a normal daylight cycle, just like you're on a spinning planet. Yet if you're doing 1 hour on, 1 hour off, it's unfavorable because the oscillator starts, then it has to reset. And so I think that's why we're seeing what we're seeing, and we're testing that hypothesis. But going back to practical application, okay, now this is where you say, Folta, you're an idiot. You're still giving 12 hours of light a day. It's just breaking it into little pieces. How are you saving anyone any energy? Okay, so what if we were to do 5 seconds on, 5 seconds off? Okay, still 12 hours a day of total illumination, same DLI, daily light integral. What if we did 5 seconds on, 6 seconds off? What if we did 5 seconds on, 10 seconds off? 5 seconds on, 20 seconds off? Now you begin to see how this might work, because we're not perturbing the oscillator, yet we're providing light energy for photosynthesis. We're just giving it in packets every so many seconds. And the plant isn't perturbed physiologically because the oscillator can't really figure out what's going on. So this is a way in which would work great in maybe being able to save some energy in a controlled environment situation by pulsing the light. And so when we did this, it worked really well. If we stay on this end where we look at kind of blue, red, and far red treatments, is you could go 5 seconds on, 5 seconds off and show a nice short seedling that's integrating. And this is hypocotyl elongation on this axis. And then this one is 5, 10, 20, 40, 160 seconds between pulses. And what you see is that whether you give 5 seconds on, 5 seconds off, or 10 seconds on, 10 seconds off, doesn't matter. And in some cases, you see sustained integration. And this is only at one fluence rate, one light intensity, that as we increase the light intensity, we actually bring this out a little further. So what this says is that the ability to sustain a light signal into a growth and development response can be done in darkness. If you apply that light, then you let it go in darkness. And we've been able to show that we've been able to get this significantly out. I mean, we've been probably 20 seconds stays about the same when we use certain fluence rates of light. So this seems like a really positive way to be able to control and modulate the growth of seedlings or plants in a controlled environment. The other thing we did, which I neglected to put in the slides here, is we did this in Arabidopsis. And so when we did this in Arabidopsis, we were able to test the photomorphogenic mutants of Arabidopsis and say, what are the light sensors that are contributing to the response? And what we were able to show is that this is primarily a phytochrome A, B, and phytochrome B, and CRY1, cryptochrome 1 mediated response. So it's responding to blue, red, and far red portions of the spectrum, a lot like we see here. And so the way to think about this is, so what we were thinking about was, okay, if we take away those receptors, we still see that they're, well, we see that they return to the normal size because of a lack of inhibition. But biology works in two directions. Usually when you look at biology, for every push, you got a pull, right? That for every inhibition, you've got something that's promoting. And these things kind of work against each other. We see this exquisitely in hormone systems where positive and negative regulators work in concert together to modulate the precision of some sort of output response. And in keeping with that idea, we started to think about what other ways might there be a pushback against this process. And so this was a really cool experiment that was done back by Shuwei Shang a couple of years ago, a visiting scientist, and was just recently repeated by an undergraduate in the lab. And this is the phototropin-1 mutant, another blue light receptor. So when we do this same experiment in the phototropin-1 mutants, we show that the light signal, and one interpretation may be that the light signal is sustained even out past 40 seconds of darkness. And still, even after 80 seconds off, you still see sustained evidence that that light signal is persisting. So it's almost like Phot-1 is the system which is shutting off the light input. You've got some that are stepping on the gas, some that are stepping on the brakes. Here, we've taken away the brakes. Why that's important is can you imagine moving into a gene editing situation here? Could we mutagenize Phot-1, knock out Phot-1, and now be able to make plants that are even better at integrating light signals? And if you don't like genetic engineering and CRISPR stuff, what if you just looked for the plants in your favorite crop that didn't bend towards light when mutagenized? Do those incorporate light signals better? And that's the hypothesis we're testing right now. So we're looking at, whereas these are driving it forward, Phot-1 appears to be pushing back. And it doesn't seem consistent with Phot-1's known roles, but no one's ever grown Phot-1, no one's ever grown plants under a flashing light at 5-second intervals before. So the take-home message on all of this is that when we put plants in a truly novel environment, which is what we're going to do in space, can we identify new trends and new ways that may be able to be translated into controlled environments here on the Earth? And I think this is a really nice example that you can do something that maybe doesn't make a lot of sense on the outside, but begin to disclose new phenomenology that not only define biology a little bit more different, but also commercial implications if it works. Because finally, if we can grow plants in a controlled environment, a closed controlled environment, and only apply the light signal once every 20 seconds, perhaps that has the ability to translate into viability in that particular industry. So that was really the new idea, is can we come up with new varieties by looking at light phenotypes of seedlings that generate new varieties that may be more amenable to use in controlled environments? And can we come up with funny ways to illuminate plants that also give us new practices? And can we put the two together? So can we select for a plant that does well in a completely foreign environment? And is that going to be a way that we can add to food security in the future, have more choices for consumers in the industrialized world, and maybe have more opportunities to explore plant biology by creating something unusual and seeing how it works in a strange environment? So that's my punchline. So so just to kind of conclude, we need to have our genetics match the environment. Just like the dachshund's pulling the dog sled, you have to have genetics that match. And that can we use seedling traits to see what's going to work in CEA? And I think we can. And then and that the traits of interest for controlled environment are very different from the field, potentially. And some of the things that we find for CEA may translate well to the field under hot conditions or conditions like Florida. And can we use unconventional delivery of light to save energy? And this idea of pulses, where we're actually investing darkness to create more phenotypes through plasticity, and at the same time, producing good consumer products by saving money, that could be potentially very nice. And so combining all this together may be the way that we solve those problems. And so with that, I will thank you and check out Talking Biotech. It took me 10 years since I started doing this stupid thing. And every week for the last 10 years. So check that out. And I guess that's everything. If you, oh, the other thing is that if you're a student or a postdoc and you want to join me as a co-host, please reach out. I'd love to have you aboard. It really works out nice. I write all the questions ahead of time, and you sound brilliant. And it's a great way to get another line in the CV. And more importantly, it's another way to dip your toe in science communication and also show evidence that you're interested in public education and extension. And for those of you who are on the job market, that one line may make a very important impact. As someone who was a former department chair, we had stacks of applicants like this. And the top this much were all very good. And the things that separated them may have been a line about how they were interested in public education and extension. So take me up on that and I'd be happy to do it. So why don't I stop there and I'll be very happy to take questions. Questions? At least, yeah. Hi. Ooh. Yeah, so C3, C4 and CAM systems, how they will respond differently. You know, I have no idea. I couldn't have guessed how they were going to respond. So yeah, I haven't gotten that sophisticated yet in thinking about or testing it. But yeah, there may be some differences. That's a good point. Yes. Yeah, so the question is, even though we heard that making something simple is better and simplifying our approaches makes more sense, here we're looking at a whole variety of different variables and complexities that make this even more challenging. How do you bring the two together? I think that's the trick though, right? How do we, and it is a good question. I think that I appreciate the idea of simplicity as well. And I've always been an underthinker of problems, and that's been my key to success. And maybe that's ultimately what where all of these complicated ideas come from, is how do we just do something different? You know, and it is a question of underthinking it. I think it is maybe it is making it simpler. It's a complicated question. Can I think about that and get back to you? I'm trying to think about it on my feet, and I'm confusing myself. Yeah. Yeah, but here's the point. So the comment was that you can make things very simple with a controlled environment. So why make it complex with lots of variables? And I think that plants are really complex. And in order to maximize the output, we have to be sensitive to those complexities and learn how to optimize our inputs to get the most appropriate output. So yeah, we can make it simple. And we can say we're going to put plants under one spectrum and grow them for 38 days and then sell it in the market. Or perhaps say, let's interject these ideas of early in development, we can get greater leaf expansion with more blue light and far red, and by limiting the red. And then as this thing grows, we'll put it under a complete spectrum. Then we're going to put it in a far red enhanced spectrum to elongate the leaves and get more dollars in the bag for the grower. And then at the end, we're going to suppress the flowering response and bolting with application of red light or whatever it is. That kind of dynamic system is something you can only do by taking apart and dissecting each part of a plant's biology. And so while I appreciate the simplicity, I think it's very true. Plants are complex, and let's play with that complexity and play into it. I think that's the better answer for me. Any other questions? Yeah. Sure. So the question is, how do you know you're making good changes? And ultimately, could you have flimsier leaves? Or, as we've shown before, massive accumulation of glucosinolates that are so awful that you can't taste it. It's so bitter. Yeah, these are all really good points. But the answer is we get to do the experiment. And I think there's a tremendous sense of job security that'll take me through retirement to sort this out. Because what is there inside a given genotype that we can squeeze out by pushing the different levers? And I think that's neat. I think that's neat. Yeah, let me go here. Yeah. Yeah, so the question is, when you're pulsing light, are you shortening the lifespan of commercial light arrays, LEDs and their associated electronics? And yeah, all these things are controlled with pulse wave modulators, and you're jamming current through these things left and right on pulses. It may shorten them, but that's for engineers to figure out. And I'm a guy with a soldering iron and enough know-how to be dangerous, and build funny things out of Raspberry Pis. So it probably does. It probably is really hard on the electronics, and it probably doesn't save as much electricity as we think because there's a little spike when you start those LEDs. So there are some considerations. So we've looked at that, but yeah, ultimately, I don't think that it would take away from the energy savings ultimately. And even if it did, energy savings are energy savings, and people, and I think that's important, is saving energy, especially, you know, and probably could argue it takes more energy to make the damn LEDs, you know, and that may be true. Yeah, good point. Oh, yeah, you just gave me something new to think about there. No, but we've been doing a lot. These are really easy experiments for undergraduates. Is, you know, take these plants and put them in some condition, and then we're going to move them to normal and see what happens. And we're seeing a lot of evidence of this. That there must be something early that happens in development, either at the level of the plastid or something epigenetic that happens during your first days of existence that shapes the way you respond to things later. And I think that kind of this also holds a potential to be applicable in controlled environments, because if we can give you this conditioning early, like blue light, first days, that you're going to flower later, that's pretty good. I think people are much happier spraying plants with photons than chemistry. And so I think this bodes well for the potential in controlled environments. Yes. Yeah, so we're not doing those experiments yet. I'm excited to think about writing to get money to do them, but I'm short on preliminary data. We've got some beautiful suggestive data that look like we're onto something, but I don't think enough to motivate a panel to throw dollars at us yet. So I've got a team of undergraduates chipping away at this as we speak. So yeah, no, they may have gone home by now. Never mind. Any other questions? All right. Got any more? Oh, okay, good. We got a couple more. Yeah. No, so have we done this experiment on strawberry or other crop genetics? Haven't really yet. We've stuck with kind of these leafy green sprouty things because they're leafy greens and sprouts because they're directly germane to the industry in controlled environments. Strawberries are too, but that's a fruit and that's a whole nother thing. I've kind of drifted out of strawberries. It was a big deal for a long time, but there's not as much industry interest, and they're more interested in ways of dealing with practical problems, and competition from Mexico, you know, and things like that are much bigger issues than can we put a funny light on it, you know. So yeah, unfortunately, yeah. There's another question. Yes. You've read my proposal for the Swiss Army plant. Yeah. Yeah, this was an idea for NASA. You know, you can't take it with you, can't take a lot of things with you. So can you take something that can agilely produce a lot of useful secondary metabolites and put them under different controllable promoters to take one plant that can give you different secondary metabolites that could be used either as antibiotics, chemotherapeutic agents potentially, or whatever. You know, could you do that? I think there's something there. I think that's a really neat idea. For those of you who are big fans of brassica domestication, you know you can take one set of genetics and tug it in a couple different directions with a few tweaks to get very different outputs. And I think we could use controllable promoters and the signals that ignite them to give us different outputs. So, yeah, it's a pretty neat idea. Anything else? Give up, end of day? I'll be around so we can talk more. But do you have another question? I thought I saw him. No, okay. Well, thank you very much. Thank you for having me over.
Yeah, I would really like to thank Kevin. I got multiple emails last night that I woke up to. He arrived in Buffalo sometime after midnight and made the drive from Buffalo to Ithaca. I think he got in Ithaca at 5 a.m., was it? So then he got here around 10. So he's a trooper, and we very much appreciate your commitment to our symposium. Well, that concludes our symposium. I'd like to thank all of you here and all of you on Zoom for attending this wonderful occasion that we've had a chance to hear these fantastic speakers. So can we get another round of applause for everyone who spoke today? And I'll keep asking for some applause. Can we thank Jason, our wonderful IT tech, who's been so helpful this year and last year. And I'd also like to thank the SIPS event coordinator, Danielle Hill. She coordinated travel with all these speakers and helped order the food and just made the committee's lives very easy. So can we give her a round of applause? And I'm going to make your hands sore. Can we also give a round of applause to the committee, Tim, Baswati, Bahiya, Shrikant, and me and everyone else. Woo, yeah. And of course, we can't forget our funding sources. So thank you, Corteva. You're amazing. And we very much appreciate all you guys do. So, when we kind of planned this committee, we wanted a wide range of topics. And so we picked a very broad theme. And I think, we heard about mushrooms, algae, lettuce, cover crops, really everything you can think of in plant breeding, and well, really not plant breeding, but in just breeding in general. And so I hope you guys can come away with, some novel ideas and thoughts for your own research. And maybe for us grad students, maybe we don't have to stick with our crops and we can venture forth to something new and exciting. Yeah, I'd like to thank you all, and please join us for happy hour and continue networking and making good friendships with everyone. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.